പാവസേ പാവൂസ് ഈ ലൈറ്റൊക്കെ ഒന്ന് ഓൺ ചെയ്തടാ, കുറച്ചുകൂടി വെളിച്ചം കിട്ടുമായിരിക്കും. അത് പുറത്തൊരായിട്ട് രണ്ടെണ്ണം ഓൺ ചെയ്തിട്ട് എന്താ കാര്യം?
Hello? Hello, Minibus. Can you hear me?
ഉടനെ തന്നെ തുടങ്ങും സാറേ. നമ്മൾ പ്രിൻസിപ്പൽ അതിനു വേണ്ടി വെയിറ്റ് ചെയ്യാണ് സാറേ. സാറേ ഉടനെ തന്നെ കേൾക്കാം സാറേ.
Okay. I was just testing.
Okay, സാറേ.
A very good afternoon to all. The most anxiously awaited occasion is finally here. On this precious day, we are witnessing a proud moment in the history of Food Science Department. I am much delighted to offer a hearty welcome to everyone. Thank you for joining us in this day. I am really honored to announce that our department has been in its 25th year. It's my privilege to welcome all to this 25th anniversary event to celebrate an important milestone in our Food Science and Technology department. This is indeed a precious moment to recall the past, thank for what we are today, and to keep ourselves motivated and energized in the years to come. Now, I would like to call upon the strength and backbone of our department, Mrs. Minnie Michael, head of the department, to deliver the welcome speech.
Respected Principal of the College, Dr. Raji Varghese Mekadan sir, respected Course Coordinator and Bursar of the College, Reverend Father George Ligalail, very respected First Coordinator of the program, Professor Thomas V. Alapat sir, our guest today for the international webinar, Dr. St.George, dear colleagues and my dear friends, good afternoon everyone. This is a solemn moment of pride and fulfillment for the Department of Food Science and Technology. My involvement with the Food Technology Department and St.George's College at Vitra started 25 years ago. And today I stand here humbled, enriched, committed because of that exact experience. The past 25 years have been punctuated with hurdles and moments of achievements. In 1996, the department had a very humble beginning with BSc Food Science and Quality Control, starting with 2 faculty members, 27 students, and 2 lab facilities. Now I am proudly able to say we have now grown in size and strength with 10 faculties, 300 students, and 6 laboratories. In 2013, we started our MSc program in food technology, and by 2018, we could start BVOC program in food technology and analysis. Our BSc program belonged to the first generation of new gen courses, and as such, the initial difficulties we encountered were many, especially regarding on the job-on training and the internship program. We do believe that the department was able to make major strides, especially in the matter of employability. The course has served as a window to the wide world of career opportunities to the students. I take this opportunity to thank our former managers, principals, and former faculty members for their valuable contributions, which helped in our journey forward. Our principal, Dr. Varghese McAdam, is a perfect captain who steers our institution in the best possible way. As an excellent administrator, endowed with great practical knowledge, he is a reservoir of energy for all our activities. Let me extend a very warm welcome to you, sir. Our bursar and coordinator of the course, Father George Puligalai, has always been a dynamic presence in the campus and lent supporting hands for the growth of our department. Further, let me extend a wholehearted welcome. A strong building needs a strong foundation. An unshakable foundation of the department was laid 25 years back by one of our eminent teachers of the college has ever seen. It's none other than our Professor Thomas B. Alapat, fondly called Alapat Sir by his colleagues and students. He was the first course coordinator of BSc Food Science and Quality program from 1996 to 99. He was a relentless academician and taskmaker and was very affectionate father figure for us. The guidance he gave and the experience we gained from him was our greatest resource. We really indebted to you, dear sir, for all your earnest help and extend a very warm welcome to you, sir. One of my greatest privileges of being a teacher was to have students with so much enthusiasm and curiosity that kept me on toes and pushed me to learn more every day to meet the seal of my students. Dr. St.George, Associate Professor in the Department of Food Science and Agricultural Chemistry, McGill University, Canada, was one among us the first batch of the students I taught. After BSc Food Science and Quality Control, he took MSc in Biotechnology from Mahatma Gandhi University, and later he worked as Junior Research Fellow at Central Food Technological Research Institute, Mysore. Thereafter, he completed PhD program from National University of Singapore. Saji, he is excellent in teaching and research, and his excellence in teaching and research have been recognized by Ministry of Education, Singapore through the Best Mentor Award. And the excellence through the continuous enterprise learning was awarded. He received the award of Gold and Silver Awards in 2015 and 16. And we appreciate you very well. And let me extend a very warm welcome to you, Dr. St.George. Now let me welcome the whole food science team, especially my colleagues who have worked tirelessly with the team spirit for the amazing journey so far. Of course, the students are the backbone of every educational institution. Now let me welcome all my dear students on this occasion. Thank you.
Thank you, Miss. Let me invite Dr. Reggie Varghese Mekaden, principal of our institution, who is very supporting and encouraging in all our activities, to speak out few words.
നമ്മുടെ കോളേജിന്റെ ഏറ്റവും ആദരണീയനായ മാനേജർ പെരിയ ബഹുമാനപ്പെട്ട അഗസ്റ്റിൻ പാലക്കിപ്പറമ്പിലച്ച, കോളേജിന്റെ കോഴ്സ് കോർഡിനേറ്ററും ബർസാറുമായ ജോർജ് പുലിയലച്ച, നമ്മളെ മോണിറ്ററിൽ കാണുന്നത് പോലെ അരുവിത്തറ കോളേജിലെ ആദ്യ കാല മുതല് നിറഞ്ഞു നിന്ന കോളേജിന്റെ ഏറ്റവും സമാരാധ്യനായ അധ്യാപകനും ഫിസിക്സ് ഡിപ്പാർട്ട്മെന്റ് മേധാവിയുമായിരുന്ന ആലപ്പാട്ട് സാറെ, തോമസ് ആലപ്പാട്ട് സാറേ, നമ്മുടെ കോളേജിലെ ഫുഡ് സയൻസിൽ നിന്ന് കടന്നുപോയി വളരെ മിടുക്കനായി അസിസ്റ്റന്റ് പ്രൊഫസറായി കാനഡയിൽ ജോലിയെടുക്കുന്ന സജി ജോർജ് സാറേ, മിനിമിസ്, പ്രിയ അധ്യാപക ശ്രേഷ്ഠരെ, പ്രിയ വിദ്യാർത്ഥിനികളെ, ഇന്ന് രണ്ടാമത്തെ പ്രോഗ്രാമാണ് സത്യം പറഞ്ഞാൽ ഫുഡ് സയൻസുമായി ബന്ധപ്പെടുത്തി കാണുവാൻ സാധിക്കുന്നത്. ഒന്ന്, വേൾഡ് ഫുഡ് ഡേയുമായി ബന്ധപ്പെടുത്തിയായിരുന്നെങ്കിൽ ഇന്ന് ഈ കലാലയത്തിലെ ഈ ഡിപ്പാർട്ട്മെന്റ് തുടങ്ങിയതിന്റെ ഒരു ജൂബിലി വർഷം കൂടി ആചരിക്കുന്ന ഒരു അവസരമാണ്. അതിൽ ഏറ്റവും വേണ്ടപ്പെട്ടവർ തന്നെ പങ്കെടുക്കുന്ന ഒരു മഹനീയ സമ്മേളനം കൂടിയാണിത് എന്ന് പറയുമ്പോൾ സന്തോഷമുണ്ട്. 25 കൊല്ലം മുമ്പ് ആലപ്പാട്ട് അച്ഛന്റെ കാലഘട്ടത്തിൽ, പ്രിൻസിപ്പൽ ആലപ്പാട്ട് അച്ഛന്റെ കാലത്തിലാണല്ലോ ഈ ഫുഡ് സയൻസ് എന്നുള്ള ഡിപ്പാർട്ട്മെന്റ് ഈ കോളേജിലെ UGCയുടെ പേരിൽ ആരംഭിക്കുന്നത്. അന്ന് മുതൽ മിനിമിസ് അതിന്റെ ചാർജായി ഉണ്ടെങ്കിൽ പോലും ആദ്യ ചുമതലക്കാരൻ ആലപ്പാട്ട് സാറായിരുന്നു. ഈ കോളേജിന്റെ വളർച്ചയിലെ എപ്പോഴും കൂടെ നിന്ന ഒരു വ്യക്തിത്വമാണ് ആലപ്പാട്ട് സാറ് എന്ന് നമ്മൾക്കറിയാം. ഞങ്ങളൊക്കെ അധ്യാപകനായി കയറുമ്പോഴേ ഒരു റോൾ മോഡലായി നമ്മുടെ മനസ്സിൽ വന്നിരുന്ന ഒരു വ്യക്തിത്വമാണ് ആലപ്പാട്ട് സാർ എന്ന് പറയുന്നത്. അപ്പോൾ ആലപ്പാട്ട് സാറിന്റെ ഒരു സാന്നിധ്യം ഈ മീറ്റിങ്ങിന് മികവ് ഉണ്ടാക്കുന്നു എന്നുള്ളത് സത്യമാണ്. 25 കൊല്ലം മുമ്പ് ആരംഭിച്ച ഈ ഡിപ്പാർട്ട്മെന്റ് അരുവിത്തറ കോളേജിന്റെ പ്രശസ്തി കേരളം ഒട്ടുക മാത്രമല്ല ലോകം മുഴുവൻ ഉണ്ടാക്കിയെടുക്കുന്നുള്ള സത്യം. ഇന്നും ഡിഗ്രി അഡ്മിഷന് ഏറ്റവും കൂടുതൽ ചോദ്യങ്ങൾ വരുന്നത് ഫുഡ് സയൻസ് ഡിപ്പാർട്ട്മെന്റിൽ അഡ്മിഷൻ കിട്ടുക എന്നുള്ളതാണ്. അന്ന് മുതൽ ഇന്ന് വരെ അത് ഭംഗിയായി നിലനിർത്താൻ സാധിച്ചു എന്നുള്ളതാണ് സത്യം. ടീച്ചർ സൂചിപ്പിച്ചതുപോലെ അന്ന് മുതൽ ഇന്ന് വരെയുള്ള എല്ലാ മാനേജർമാരും, അന്ന് മുതൽ ഇന്ന് വരെയുള്ള എല്ലാ പ്രിൻസിപ്പൽമാരും ഈ ഡിപ്പാർട്ട്മെന്റിനെ അതുപോലെ തന്നെ കരുതലോടെ കണ്ടതുകൊണ്ടായിരിക്കാം ഇപ്പോഴും ഇത് വളരെ ഭംഗിയായി മുന്നോട്ട് പോകാൻ സാധിക്കുന്നത്. ഇന്ന് സജി ജോർജ് സാറിനെ കുറിച്ച് കേട്ടു. അദ്ദേഹത്തിന്റെ ക്ലാസ് കേൾക്കുന്നുണ്ട്. തീർച്ചയായിട്ടും ഈ കലാലയത്തിൽ 25 കൊല്ലം മുമ്പ് പഠിച്ച് ഇറങ്ങിപ്പോയപ്പോൾ അതിന്റെ ഒരു ഗ്രഹാതുരത്വം അദ്ദേഹത്തിന് അനുഭവപ്പെടുന്നുണ്ടായിരിക്കും എന്ന് തന്നെയാണ് എനിക്ക് തോന്നുന്നത്. ടീച്ചറിനെ നേരിട്ട് കാണുന്നതിനും സംസാരിക്കുന്നതിനും എല്ലാം സാധിച്ചിട്ടുണ്ട്. ഈ കലാലയം അദ്ദേഹത്തിന്റെ കലാലയമാണ്. നമ്മുടെ ഒരു സ്ഥാപനത്തെ കുറിച്ച് പൊതുവെ നമ്മൾ പറയുമ്പോൾ ആ സ്ഥാപനം മികച്ചതായി മാറുന്നത് ആ സ്ഥാപനത്തിൽ പഠിച്ചിറങ്ങിയ വിദ്യാർത്ഥികളുടെ കഴിവിനെ ആശ്രയിച്ചും അവര് ഏതെല്ലാം സ്ഥാനമാനങ്ങൾ അലങ്കരിക്കുന്നു എന്നുള്ളതിനെ കുറിച്ചുമാണ്. അത്തരത്തിൽ അഭിമാനം നൽകുന്ന വ്യക്തിത്വമാണ് സജി ജോർജ് സാറൻ. സജി ജോർജ് സാറിന്റെ സാന്നിധ്യം സന്തോഷം നൽകുന്നു. ഡിപ്പാർട്ട്മെന്റിനെ വളരെ കാര്യക്ഷമതയോടെ നയിക്കുന്ന മിനിമിസിനെ പ്രത്യേകം അഭിനന്ദിക്കുന്നു. ഇതെല്ലാം മിനിമിസ് ഓർത്ത് ഇരുന്ന് കോർത്തിണക്കി ഉണ്ടാക്കുന്ന പ്രോഗ്രാമാണ്. ആലപ്പാട് സാറിനെയും ഇവരെല്ലാം ഓർത്ത് നിങ്ങൾ പ്രോഗ്രാം സംഘടിപ്പിച്ചതിനെ പ്രത്യേകം അഭിനന്ദിക്കുന്നു. എപ്പോഴും ഈ കോളേജിലെ ഫുഡ് സയൻസ് ഡിപ്പാർട്ട്മെന്റ് മികവുറ്റതാണ്, അത് ഭാവിയിലും മികവുറ്റതാണ് എന്ന് തെളിയിക്കപ്പെടുവാൻ ഈ മീറ്റിംഗുകൾ കാരണമാകട്ടെ എന്ന് ആശംസിച്ചുകൊണ്ട് നിർത്തുന്നു. നന്ദി.
Thank you, sir. It gives me immense pleasure to invite respected bursar and course coordinator. He is always have a keen interest and promote us in good initiation. So I humbly request Father George Kalai to deliver few words.
ഏറ്റവും ആദരണീയനായ കോളേജിന്റെ പ്രിൻസിപ്പൽ ഡോക്ടർ റെജി വർഗീസ് മേക്കാടൻ സാറേ, ഇന്നത്തെ വിശിഷ്ട അതിഥി കോളേജിലെ മുൻ അധ്യാപകൻ കൂടിയായ പ്രൊഫസർ തോമസ് വി ആലപ്പാട്ട് സാറേ, നമ്മളുടെ ഒരു വെബിനാർ സീരീസ് തന്നെയാണ് നമ്മൾ ഇന്ന് ആരംഭിക്കുന്നത്. ഈ സീരീസിന്റെ ആദ്യ ടോക്ക് അതിനുവേണ്ടി റിസോഴ്സ് പേഴ്സണായി എത്തിയിരിക്കുന്ന ഈ ഡിപ്പാർട്ട്മെന്റിലെ ആദ്യത്തെ സ്റ്റുഡൻസിൽ ഒരാളായ ഡോക്ടർ സജി ജോർജ്, വകുപ്പ് മേധാവി മിനിമിസ്, മറ്റ് അധ്യാപകരെ, ഏറ്റവും പ്രിയപ്പെട്ട വിദ്യാർത്ഥികളെ, ഈ സെമിനാറിൽ പങ്കെടുക്കുന്ന മറ്റ് വിശിഷ്ട വ്യക്തികളെ, ഈ ഡിപ്പാർട്ട്മെന്റിന്റെ ഫുഡ് സയൻസ് ഡിപ്പാർട്ട്മെന്റിന്റെ 25ാം വാർഷികം ജൂബിലി ആഘോഷിക്കുകയാണ്. അതിനോട് ബന്ധപ്പെടുത്തിയാണ് ഈ മീറ്റിംഗ് ഇന്നിപ്പോൾ ഇവിടെ സംഘടിപ്പിക്കപ്പെടുന്നത്. അരുവിത്തറ കോളേജിനെ സംബന്ധിച്ചിടത്തോളം വളരെ അഭിമാനം നിറഞ്ഞു നിൽക്കുന്ന നിമിഷമാണിത്. ഫുഡ് സയൻസ് ഡിപ്പാർട്ട്മെന്റിന്റെ ജൂബിലി ആഘോഷിക്കുമ്പോൾ ഈ ഡിപ്പാർട്ട്മെന്റ് കോളേജിന് നൽകിയ വലിയ സേവനങ്ങളെ നന്ദിയോടുകൂടെ ഓർക്കുകയാണ്. അരുവിത്തറ കോളേജ് കേരളം മുഴുവൻ അറിയപ്പെടുന്നത് ഈ ഡിപ്പാർട്ട്മെന്റിലൂടെയാണെന്ന് ഒരുപക്ഷെ പറയാം. ഞാൻ ഈ നാളുകളിലൊക്കെ ഫുഡ് സയൻസ് ഡിപ്പാർട്ട്മെന്റിലേക്കും മറ്റ് ഡിപ്പാർട്ട്മെന്റുകളിലേക്കും കുട്ടികളെ മാനേജ്മെന്റ് കോട്ട അഡ്മിഷൻ നടത്തുന്ന തിരക്കിലായിരുന്നു. അപ്പോൾ ആളുകൾ തേടി ഏറ്റവും കൂടുതൽ കോഴ്സ് പഠിക്കാനായിട്ട് ആഗ്രഹിച്ച് ഇവിടെ വരുന്നത് ഫുഡ് സയൻസ്, BSc ഫുഡ് സയൻസ്, MSc ഫുഡ് സയൻസ് എന്നീ വിഷയങ്ങളാണ്. പലപ്പോഴും അതിൽ അഭിമാനം തോന്നിയിട്ടുണ്ട്. കേരളത്തിന് പുറത്ത്, കേരളത്തിനുള്ളിൽ മാത്രമല്ല, കേരളത്തിന് പുറത്ത് ഇന്ത്യയിലെ മറ്റ് സംസ്ഥാനങ്ങളിൽ നിന്നും അതുപോലെ തന്നെ വിദേശ രാജ്യങ്ങളിൽ നിന്നും മൈഗ്രൻസ് ആയി ജീവിക്കുന്ന ഗൾഫ് റീജിയനിലുള്ള ആൾക്കാര് ഈ വിഷയത്തിന് വേണ്ടി പലപ്പോഴും എന്നെ വിളിച്ച് എൻക്വയറി നടത്തുകയും അഡ്മിഷൻ ആഗ്രഹിച്ച് സംസാരിക്കുകയും ചെയ്യാറുണ്ട്. അത്തരത്തിൽ കോളേജിന് പേരും പെരുമയും നൽകുന്ന ഒരു ഡിപ്പാർട്ട്മെന്റ് ആണ് ഫുഡ് സയൻസ് എന്ന് പറയുന്നു. അതുപോലെ തന്നെ കോളേജിൽ ഏറ്റവും അക്കാഡമിക് ആയിട്ടുള്ള നല്ല സ്കോറുള്ള കുട്ടികൾ വരുന്നത് ഫുഡ് സയൻസ് ഡിപ്പാർട്ട്മെന്റിലാണ്. അതുകൊണ്ട് കോളേജിന്റെ അച്ചടക്കവും അതുപോലെ തന്നെ റിസൾട്ടും നല്ലതാകാൻ സഹായിക്കുന്നു. ഇവിടെ പഠിച്ച വിദ്യാർത്ഥികൾ, അതല്ലെങ്കിൽ ഇവിടെ പഠിക്കുന്ന വിദ്യാർത്ഥികൾക്ക് ഒരു ഭൂരിഭാഗം കുട്ടികൾക്കും നല്ല തൊഴിൽ ലഭിക്കുന്നു എന്നുള്ളത് ഈ കോളേജിനെ സംബന്ധിച്ചിടത്തോളം സന്തോഷമുള്ള കാര്യമാണ്. അത് ഈ സമയം ഞാൻ സ്മരിക്കുകയാണ്. വിവിധ മേഖലകളിലായി ലോകമെമ്പാടും അരുവിത്തറ കോളേജിൽ ഫുഡ് സയൻസ് പഠിച്ച വിദ്യാർത്ഥികൾ നല്ല രീതിയിൽ ജോലി ചെയ്യുന്നു, സന്തോഷത്തോടെ ജീവിക്കുന്നു എന്ന് പറയുന്നതിൽ അഭിമാനമുണ്ട്. ഇങ്ങനെയൊക്കെ അപ്പൊ ഇത്തരത്തിൽ കോളേജിനെ വളർത്തിയ ഈ ഡിപ്പാർട്ട്മെന്റിൽ ഇതുവരെയും വർക്ക് ചെയ്ത എല്ലാ അധ്യാപകരെയും അതുപോലെ തന്നെ ഇപ്പോൾ പ്രവർത്തിച്ചുകൊണ്ടിരിക്കുന്ന മിനിമിസിന്റെ നേതൃത്വത്തിൽ പ്രവർത്തിച്ചുകൊണ്ടിരിക്കുന്ന എല്ലാ അധ്യാപകർക്കും ഈ നിമിഷം ഞാൻ പ്രത്യേകം നന്ദി പറയുകയാണ്. ഈ ആഘോഷവേളയിൽ, ഈ ജൂബിലി വേളയിൽ പറയേണ്ട മറ്റൊരു കാര്യം കൂടിയുണ്ട്. ഇനി മുന്നോട്ട് ഈ കോളേജിന് വളർച്ച കൊണ്ടുവരേണ്ടതും ഈ ഡിപ്പാർട്ട്മെന്റ് തന്നെയായിരിക്കണം. മിനി ടീച്ചർ പറഞ്ഞു, വളരെയേറെ നമ്മൾ മുന്നോട്ട് പോയിട്ടുണ്ട്, ഇനിയും വളരെ മുന്നോട്ട് പോകാനുണ്ട്. ഫുഡ് സയൻസ് ഡിപ്പാർട്ട്മെന്റ് അരുവിത്തറ കോളേജിലെ വലിയൊരു റിസർച്ച് ഡിപ്പാർട്ട്മെന്റായി തീരട്ടെ എന്ന് പ്രതീക്ഷിക്കുകയാണ്, ആഗ്രഹിക്കുകയാണ്. ഈ വെബിനാറിൽ പങ്കെടുക്കുന്ന എല്ലാവരെയും ഞാൻ നിങ്ങളുടെ സാന്നിധ്യത്തെ വിലമതിക്കുന്നു. പ്രത്യേകിച്ച് ഈ ഡിപ്പാർട്ട്മെന്റിന് അങ്ങേയറ്റം സേവനം ചെയ്ത ആദ്യകാല കോർഡിനേറ്ററായ പ്രൊഫസർ തോമസ് വി ആലപ്പാട്ട് സാറിനെ എനിക്ക് മുൻപേ അറിയാം. സാറിന്റെ സാന്നിധ്യത്തെ വിലമതിക്കുന്നു. സാർ ഇപ്പോൾ ഞങ്ങളോടുകൂടെ ആയിരിക്കുന്നതിൽ വലിയ സന്തോഷമുണ്ട്. പ്രത്യേകം നന്ദി പ്രകാശിപ്പിക്കുകയാണ്. അതുപോലെ തന്നെ ഡോക്ടർ സജി ജോർജ് ഈ കോളേജിലെ ആദ്യ ബാച്ചിലെ സ്റ്റുഡന്റ് ആണ്. എന്നാൽ ഇപ്പോൾ ഉയരങ്ങളില് വളരെ മഹത്തായ രീതിയിൽ കാനഡയിൽ സേവനം ചെയ്യുന്നു. അദ്ദേഹം ഇന്ന് ഞങ്ങളോടുകൂടെ ആയിരിക്കുന്നതിൽ ഞങ്ങൾക്ക് അതിയായ സന്തോഷമുണ്ട്. താങ്കളുടെ സാന്നിധ്യത്തെ ഞങ്ങൾ വിലമതിക്കുന്നു. ഈ എല്ലാവർക്കും നന്മകളും ആശംസകളും നേർന്നുകൊണ്ട് എന്റെ വാക്കുകൾ അവസാനിപ്പിക്കുന്നു. താങ്ക് യു.
Thank you, Father. Professor Thomas V. Alapat, Head of the Department of Physics till 1999 and first coordinator of BSc Food Science and Quality Control for the first 3 years from 96 to 99. It is our privilege to invite Professor Thomas V. Alapat for sharing his experience.
സാറേ, ആലപ്പാട്ട് സാറേ, ഒന്ന് അൺമ്യൂട്ട് ചെയ്യാമോ? സാർ പറയുന്നത് കേൾക്കുന്നില്ലേ? അത് മ്യൂട്ട് അൺമ്യൂട്ട് ആണോ? സാറേ, അൺമ്യൂട്ട് ബട്ടണിൽ ഒന്നുകൂടെ പ്രസ് ചെയ്തിട്ട് സംസാരിക്കാമോ? കേൾക്കുന്നില്ലല്ലോ. അൺമ്യൂട്ട് ചെയ്യാമോ സാറേ?
ഹലോ, ഇപ്പൊ കേൾക്കാമോ?
കേൾക്കാം.
ആദരണീയനായ മാനേജർ അച്ഛ, ബഹുമാന്യനായ കുഞ്ഞുകലയിൽ അച്ഛ, ബഹുമാന്യനായ പ്രിൻസിപ്പൽ റെജി സാറെ, മിനി ടീച്ചർ ഡോക്ടർ റെജി ജോർജ്, ബഹുമാന്യരായ മറ്റു അധ്യാപകരെ, പ്രിയപ്പെട്ട സുഹൃത്തുക്കളെ, നിങ്ങളെ മുൻകാല വിദ്യാർത്ഥികൾ എന്ന് സംബോധന ചെയ്യുന്നതിനേക്കാൾ സുഹൃത്തുക്കൾ എന്ന് സംബോധന ചെയ്യുന്നതാണ് കൂടുതൽ ശരി എന്ന് തോന്നിയത് കൊണ്ടാണ് നിങ്ങളെ സുഹൃത്തുക്കൾ എന്ന് സംബോധന ചെയ്തത്. അരുവിത്തറ കോളേജിലെ ഫുഡ് സയൻസ് ആൻഡ് ക്വാളിറ്റി കൺട്രോൾ എന്ന കോഴ്സിൻ്റെ ആദ്യ ബാച്ചിലെ വിദ്യാർത്ഥികളുടെ ഒരു കൂട്ടായ്മ സംഘടിപ്പിക്കുന്നുണ്ട് എന്ന് മിനി ടീച്ചർ പറഞ്ഞപ്പോൾ എനിക്ക് വളരെയേറെ സന്തോഷം തോന്നി. കാരണം വളരെയേറെ വർഷങ്ങൾക്ക് ശേഷമാണ് ഒരു ഓൺലൈനായിട്ടാണെങ്കിലും നിങ്ങളെയൊക്കെ കാണാൻ സാധിക്കുന്നത്. ഏതായാലും ഈ കൂട്ടായ്മ സംഘടിപ്പിക്കുന്ന കാര്യത്തിൽ ഇതിന്റെ പുറകിൽ പ്രവർത്തിച്ച കേൾക്കുന്നില്ലേ? Can you hear? കേൾക്കാവോ?
കേൾക്കാം സാറേ, കേൾക്കാം. സാറ് പറഞ്ഞോളൂ.
ഇതിന്റെ പുറകിൽ പ്രവർത്തിച്ച എല്ലാവരെയും, മിനി ടീച്ചറാണ് ഇതിന്റെ ലീഡ് എടുത്തതെന്ന് എനിക്കറിയാം. മിനി ടീച്ചറെയും ഇതിന്റെ പുറകിൽ പ്രവർത്തിച്ച എല്ലാവരെയും അവരോട് പ്രത്യേകം നന്ദി ഈ അവസരത്തിൽ രേഖപ്പെടുത്തുകയാണ്. ഫുഡ് സയൻസിലെ ആദ്യ ബാച്ചിലെ വിദ്യാർത്ഥികൾ എന്ന് പറയുമ്പോൾ പെട്ടെന്ന് എൻ്റെ ഓർമ്മ 1996 അരുവിത്തറ കോളേജും അന്ന് അവിടെ ആരംഭിച്ച ഫുഡ് സയൻസ് എന്ന കോഴ്സിനെ കുറിച്ചുമാണ്. നമ്മുടെ യൂണിവേഴ്സിറ്റി ഗ്രാൻസ് കമ്മീഷൻ UGC 95-96 വർഷങ്ങളിൽ ജോബ് ഓറിയൻറ്റഡ് ആയിട്ടുള്ള കോഴ്സുകൾ അനുവദിക്കുന്നുണ്ട് എന്ന് മാധ്യമങ്ങളിൽ കൂടെ മനസ്സിലായപ്പോൾ അന്നത്തെ പ്രിൻസിപ്പല് ബഹുമാനപ്പെട്ട ആലപ്പാട്ട് കുന്നേലച്ഛൻ 2 കോഴ്സുകൾക്ക് വേണ്ടി അപേക്ഷിച്ചു. അതിനെ തുടർന്ന് ഞാനും ലോപ്പസാറും കൂടെ ബാംഗ്ലൂർ പോയി UGC പ്രതിനിധികളുമായിട്ട് സംസാരിച്ചു. ആ സംസാരത്തിൻ്റെ അല്ലെങ്കിൽ ആ ചർച്ചയുടെ ഫലമായിട്ട് ഒരു കോഴ്സ് നമുക്ക് അനുവദിച്ച് കിട്ടി. ദിവസങ്ങൾക്ക് ശേഷമാണ് അറിഞ്ഞത് അത് ഫുഡ് സയൻസ് ആൻഡ് ക്വാളിറ്റി കൺട്രോൾ എന്ന കോഴ്സാണെന്ന്. ഇത് ഒരു UGC സ്പോൺസേഡ് കോഴ്സ് ആയതുകൊണ്ട് ഇതിന് ഒരു പ്രോപ്പർ സിലബസ് പോലും അന്ന് MG യൂണിവേഴ്സിറ്റിയിൽ ഇല്ലായിരുന്നു. ഇതിനിടയിൽ കോഴ്സ് കോർഡിനേറ്ററായിട്ട് ഞാൻ ചാർജ് എടുത്തു. ഇതിൻ്റെ തലേ വർഷം, അതായത് 1995 ചങ്ങനാശ്ശേരി NSS കോളേജിലും കോട്ടയം BCM കോളേജിലും ഈ കോഴ്സുകൾ ആരംഭിച്ചിട്ടുണ്ടായിരുന്നു. ഇതിനെക്കുറിച്ച് കൂടുതൽ അറിയാൻ വേണ്ടി അവിടെ ചെന്നപ്പോൾ നമുക്ക് ആവശ്യമുള്ള ഒരു ഇൻഫർമേഷനും തരാൻ അല്ലെങ്കിൽ നമുക്ക് ലഭിച്ചില്ല. പിന്നെ അവസാനം കോയമ്പത്തൂരുള്ള വിനാശലിംഗം ഡീംഡ് യൂണിവേഴ്സിറ്റിയിൽ പോയിട്ടാണ് സിലബസിനെ കുറിച്ച് ഏകദേശം ഒരു ധാരണ നമുക്ക് കിട്ടിയത്. അതിനിടയിൽ നമ്മൾ ഈ കോഴ്സിനെ കുറിച്ച് അഡ്വർടൈസ് ചെയ്തു. മിനി ടീച്ചർ ഇവിടെ ഒരു കോളേജിലൊരു അധ്യാപികയായിട്ട് ജോയിൻ ചെയ്തു. മാത്രമല്ല ആ വർഷം തന്നെ കോളേജ് അഡ്മിഷൻ നടന്നു. നിങ്ങളെല്ലാവരും ഇവിടെ വന്ന് ചേരുകയും ചെയ്തു. അന്ന് ഈ കോഴ്സിൻ്റെ ഭാവി എന്തായിരിക്കും, ഇത് എത്ര കാലം ഈ കോഴ്സ് തുടർന്നു കൊണ്ട് പോകാൻ സാധിക്കും, ഇത് ഒരു ടെർമിനൽ കോഴ്സ് ആയതുകൊണ്ട് ഈ കോഴ്സ് പാസ്സായവർക്ക് പിന്നെ PG കോഴ്സിന് പഠിക്കാൻ അനുവാദം കിട്ടുമോ എന്നുള്ള ഒരുപാട് ആശങ്കകൾ ഞങ്ങളുടെ മനസ്സിലുണ്ടായിരുന്നു. അതൊന്നും നിങ്ങളുമായിട്ട് അന്ന് പങ്കുവച്ചില്ല എന്ന് മാത്രം. ഇത് ഒരു പുതിയ കോഴ്സ് ആയതുകൊണ്ട് ഇതിൻ്റെ ബാലാരിഷ്ടതകൾ വളരെയേറെയായിരുന്നു. ഞാൻ ആ പഴയ കാര്യത്തിലിട്ട് ഒരു കുറച്ച് സമയം ഒന്ന് ശ്രദ്ധിക്കുമെന്ന് വിചാരിക്കുന്നു. ഈ യൂണിവേഴ്സിറ്റി എക്സാമിനേഷന് ക്വസ്റ്റ്യൻ പേപ്പർ സെറ്റ് ചെയ്യുക, അതുപോലെ ആ പേപ്പറുകൾ വാല്യൂ ചെയ്യുക, ഇതിന് ക്വാളിഫൈഡ് ആയിട്ടുള്ള ഫാക്കൽറ്റിയെ കണ്ടുപിടിക്കുക, ഈ കാര്യത്തിലെല്ലാം യൂണിവേഴ്സിറ്റിയെ സഹായിക്കാൻ വേണ്ടി പല പ്രാവശ്യം യൂണിവേഴ്സിറ്റിയിലേക്ക് പോകേണ്ടി വന്നിട്ടുണ്ട്. മറ്റൊരു പ്രശ്നം, ഇതിൻ്റെ എക്സാമിനേഷൻ നടത്തിക്കുക, അതിന് സ്ലോട്ട് കിട്ടണം, അതുപോലെ തന്നെ ഇതിൻ്റെ റിസൾട്ട് പബ്ലിഷ് ചെയ്യിക്കണം. ഇതൊക്കെ നമ്മുടെ ഭാരമായിട്ട് വന്നു. അല്ലെങ്കിൽ നമ്മൾ ഇതിനു വേണ്ടിയിട്ട് യൂണിവേഴ്സിറ്റി ഓഫീസിൽ കയറി ഇറങ്ങേണ്ടി വന്നു. മറ്റൊരു പ്രശ്നം വന്നത് ജോബ് ഓൺ ട്രെയിനിങ്ങിനെ സംബന്ധിച്ചായിരുന്നു. അതായത് നിങ്ങൾ ജോബ് ഓൺ ട്രെയിനിങ്ങിന് പോകുമ്പോൾ സുരക്ഷിതമായിട്ടുള്ള സ്ഥലങ്ങൾ, നമുക്ക് പ്രയോജനപ്രദമായിട്ടുള്ള സ്ഥാപനങ്ങൾ, ഇവയെ കണ്ടുപിടിച്ച് ഇവയിൽ അഡ്മിഷൻ കിട്ടുന്നതിന് വേണ്ടി പലപ്പോഴും രാഷ്ട്രീയക്കാരെയും രാഷ്ട്രീയ തലത്തിൽ പ്രവർത്തിക്കുന്നവരെയും സാമൂഹ്യ തലത്തിൽ പ്രവർത്തിക്കുന്നവരെയും സാമ്പത്തിക തലത്തിൽ പ്രവർത്തിക്കുന്നവരെയൊക്കെ പല പ്രാവശ്യം കാണേണ്ടി വന്നിട്ടുണ്ട്. മറ്റൊരു പ്രശ്നം എൻകൗണ്ടർ ചെയ്തത് സബ്ജക്റ്റുകൾ, അതായത് സബ്സിഡറി സബ്ജക്റ്റുകളായിട്ട് നിങ്ങൾക്കറിയാം, പഠിച്ചപ്പോൾ ചില സബ്ജക്ട്സ് ഉണ്ടായിരുന്നു. ആ ക്ലാസ്സുകൾ ആര് ഹാൻഡിൽ ചെയ്യും, ആരെടുക്കും, അങ്ങനെയുള്ള ചില പ്രശ്നങ്ങളുണ്ടായിരുന്നു. ഏതായാലും മറ്റു ഡിപ്പാർട്ട്മെൻറ്റുകാരുടെ സഹായത്തോടും സഹകരണത്തോടും കൂടി ആ പ്രശ്നങ്ങളൊക്കെ അതിജീവിക്കുവാൻ സാധിച്ചു എന്ന കാര്യം ഇപ്പോൾ വളരെ സന്തോഷത്തോടു കൂടി ഓർക്കുകയാണ്. അതിലേറെ സന്തോഷം എന്നെ സംബന്ധിച്ചിടത്തോളം തരുന്നൊരു കാര്യം, അല്ലെങ്കിൽ അന്നത്തെ വിദ്യാർത്ഥികളായ അല്ലെങ്കിൽ അധ്യാപകരായ ഞങ്ങളെ സംബന്ധിച്ചിടത്തോളം ഒരു കാര്യം, 99 റിസൾട്ട് വന്നപ്പോൾ ഏറ്റവും നല്ലൊരു റിസൾട്ട് ഞാൻ റെജി ജോർജിനെ പ്രത്യേകം അഭിനന്ദിക്കുകയാണ്. യൂണിവേഴ്സിറ്റിയിലെ ഫസ്റ്റ് റാങ്ക് അന്ന് റെജി ജോർജ് വാങ്ങിച്ചു. മാത്രമല്ല മറ്റെല്ലാ വിദ്യാർത്ഥികളും ഏറ്റവും ഉയർന്ന നിലവാരത്തിലുള്ള ഒരു റിസൾട്ട് അന്ന് വാങ്ങിച്ചു. അത് ആ 3 വർഷത്തെ ഞങ്ങളുടെ കഠിനാധ്വാനത്തിന് അവർ തന്നിട്ടുള്ള ഏറ്റവും വലിയ സമ്മാനമായിട്ട് ഞാൻ ഈ അവസരത്തിൽ കരുതുകയാണ്. പിന്നെ മറ്റൊരു കാര്യം, സാധാരണഗതിയിൽ മാതാപിതാക്കളെ സംബന്ധിച്ചിടത്തോളം നിങ്ങളൊക്കെ മാതാപിതാക്കളായി കാണും എന്ന് ഞാൻ കരുതുന്നു. മാതാപിതാക്കളായി കഴിയുമ്പോൾ, അല്ലെങ്കിൽ അവർക്ക് ഏറ്റവും സന്തോഷം തരുന്ന ഒരു കാര്യം, ഒരു മുഹൂർത്തം, അവരുടെ മക്കളൊക്കെ വലിയ നിലയിൽ, അവരെക്കാൾ ഉയർന്ന നിലയിലെത്തുമ്പോഴാണ്. അത് ഏത് മാതാവിൻ്റെയും പിതാവിൻ്റെയും ഒരു വലിയ ലക്ഷ്യമാണ്. അതുപോലെ തന്നെയാണ് അധ്യാപകരുടെ ശിഷ്യന്മാർ, അവർ പഠിപ്പിച്ചവരെ സംബന്ധിച്ചിടത്തോളം അവർ ഉയർന്ന നിലയിലെത്തുക എന്നുള്ളത് ഏത് അധ്യാപകൻ്റെയും ഒരു ആഗ്രഹമാണ്. എന്നെ സംബന്ധിച്ച് പറഞ്ഞാൽ, ഞാൻ നിങ്ങൾക്ക് അക്കാദമിക വിഷയങ്ങളൊന്നും പഠിപ്പിച്ചിട്ടില്ലെങ്കിലും ആ 3 കൊല്ലം നിങ്ങളുടെ കൂട്ടത്തിലുണ്ടായിരുന്നു. നിങ്ങളെല്ലാവരും ഇന്ന് വളരെ ഉയർന്ന നിലയിൽ എത്തിയിരിക്കുന്നു എന്നറിയുമ്പോൾ എനിക്ക് വളരെ വളരെ സന്തോഷമുണ്ട്. എനിക്ക് മാത്രമല്ല, മിനി ടീച്ചറിനും മറ്റു അധ്യാപകർക്കും ഉണ്ട് എന്നെനിക്കറിയാം. ഇന്ന് നേരത്തെ അച്ഛൻ പറഞ്ഞ മാതിരി ഏറ്റവും കൂടുതൽ ഡിമാൻഡുള്ള ഒരു കോഴ്സായിട്ട് ഈ ഫുഡ് സയൻസ് ആൻഡ് ക്വാളിറ്റി കൺട്രോൾ എന്ന കോഴ്സ് വന്നിട്ടുണ്ടെങ്കിൽ അതിന് ബന്ധപ്പെട്ട എല്ലാവരോടും ഞാൻ പ്രത്യേകം നന്ദി പറയുന്നു. ഇവിടുന്ന് കടന്നുപോയ 99-ലും അതിനുശേഷവും കടന്നുപോയിട്ടുള്ള പല വിദ്യാർത്ഥികളുമായിട്ട് ഇപ്പോഴും അക്കാദമികമായിട്ട് ബന്ധപ്പെടാറുണ്ട്. അപ്പോൾ ഏതായാലും ഈ കടന്നുപോയ എല്ലാ വിദ്യാർത്ഥികളും അവരെല്ലാം ഉയർന്ന നിലവാരത്തിലെത്തിയതിൽ അവർ വളരെയേറെ സന്തോഷിക്കുകയും അവർക്ക് എല്ലാ നന്മകൾ നേരുകയും ചെയ്തുകൊണ്ട് ഞാൻ എൻ്റെ വാക്കുകൾ ഉപസംഹരിക്കുന്നു. നന്ദി, നമസ്കാരം.
Thank you, sir. Dr. St.George, our eminent alumnus, working as Associate Professor, Canada Research Chair in Sustainable Nanotechnology for Food and Agriculture, McGill University, Canada. With great honor and respect, I invite our first batch student to share his experience in this 25th anniversary. Over to you, sir.
Hello, can you hear me?
Kelkam, sir. Kelkam.
Hello.
Kelkam.
Okay. Very good, thank you. A very good afternoon, everyone, and respected principal, course coordinator, and manager sir, principal sir, and most beloved Alapad sir and Minimis, and all the students who are listening to me. I'm extremely happy, I should say. I'm extremely excited to come back to my alumni and interact with you all. I should say, like, I didn't know the background of how we got this course, how it began. And Alapad sir mentioned about the pain behind it and how he managed to run. I knew that he was running around all the time to get us the best of the best. And I believe one of the reasons why this course has been progressing so well and it's on high demand is because of those efforts that he put, the foundation that he put for this course. And I can't overemphasize how the teachers, especially Minimis, at the beginning of the course, she, spent time for us in developing the course, the curriculum, as well as in making sure that we got the best of the best in terms of our internships. Like, we've been to different industries at that time, and it actually gave us much more confidence. And for me particularly, it was, of course, a beginning of a fruitful and meaningful journey. Now I am an associate professor at the Food Science and Agriculture Chemistry Department of McGill University. And I should say that journey, the first step on that journey began from the St.George's College at Vitra. So I'm really happy for that. I'm thankful to, and I can only look back with immense gratitude for all these great work and the hardships and all this mentoring that was done through our teachers, and especially Alapad sir. Alapad sir used to give us a lot of advice. As he mentioned, he didn't teach us. That's not true. I think he taught us 1 or 2 courses in physics and in his own style. And we all remember fondly about his teaching styles and the conversations that we had with him during our trips, like our industrial visit and those kind of trips that we made. Those are valuable memories. We share that memories whenever we get together in a WhatsApp platform, for example. Now, one of the advices he gave me was, I think to our friends, one day was, he said, you may grow much higher than all your current teachers, but one thing you cannot beat us, and that is you will never outgrow our age. But when I actually look at the flyer, I thought maybe I may not have grown up in my professional, but at least I'm in par with Alapad sir in terms of age, I think. And so from the photo, it looked like we both share the same type of head or baldness. And so I'm really happy to tell you that, sir, you are still young. You're keeping yourself young, but we are growing. Okay, so with that, I will. It is my pleasure to share with you all my little bit of research, what I'm doing, and maybe what I'm hoping is it will be a motivation for you to look. To be bold, to look up, and to say that you all are competent to achieve greater heights. I won't claim that I achieved something great, but I'm sure you all have the potential to go grow and go beyond. So I call upon all of you to take home the message from me is that if I can do it, you can also do that. So I will be happy to share my studies or my endeavor here in Canada and over the past couple of years. So let me know when can I start the presentation or if there is any more proceedings. Thank you, Anando.
Hello, Saji. You can start with your presentation.
Yes.
Now. There is no other problem.
Okay, great. Okay, great. So
It's okay.
in that case, I will share my slides. So all right. So let me go to presentation now. It takes some time to load the content, so
Okay.
I hope it works well. This is the second time I'm using this platform for giving a talk. So being good. Let me see. Seems like I need permission from you to share my screen. So whoever is hosting that meeting, can you please do that?
Sure, sir. Please ask the permission.
So you have? Okay, so let me see. So let me know if you can see my presentation now.
Sorry, sir. It will come soon.
Okay. I believe you can see my presentation now. Is it?
No, sir. No, sir.
Yes?
You can present now. You can give a present now switch in the side.
Yeah, I'm trying that. Yeah, that's what I yeah, in my window it shows it is shared. Let me do it again one more time. Now I'm trying that in my window. What I see is you must grant permission in order to screen share. grant permission? How do I grant permission? I'm clicking the present now intro, like we had tried before, like a couple of days ago. It okay. If I can't present, I mean, I can't give the slides. I will try to talk.
Sir, present now എന്ന് പറഞ്ഞിട്ടൊരു ഇതുണ്ട്. അതിൽ ക്ലിക്ക് ചെയ്ത് കഴിഞ്ഞാൽ then it will appear in a window.
No, I know. So can you see now? I've been trying that. I think it is coming now.
Okay.
Can you see my screen now?
Sorry, sir. ഇതുവരെ അത് എത്തിയിട്ടില്ല. ഓക്കെ, അത് വരുന്നുണ്ടെന്ന് തോന്നുന്നു. സാറിന്റെ രണ്ട് പ്രൊഫൈൽ ഇപ്പൊ കാണാം.
Wait for a minute.
സാറിന്റെ പ്രൊഫൈൽ ഇപ്പൊ കാണാം. ഇപ്പൊ ഓക്കെ ആയി. സാറിന് okay, right.
Very good.
Right.
Okay.
Okay.
So I believe what you are seeing is my first slide, and sorry for the trouble. And this is on a daily basis. When we are using this electronic platform to teach or to give a lecture. But anyway, to all you students, it is my pleasure to share with you some of my research at McGill. So as you may know, I'm working on nanotechnology in food, and the term nanotechnology may be something new to many of the students who are taking like food science. So I thought it is a good opportunity to introduce the discipline of nanotechnology, and also bring to your attention in future, because you will be dealing with issues or applications of nanotechnology in food, and it will be a great opportunity to know what nanotechnology is, and to be confident enough to move on for a future interviews. But let me introduce where I am from. Right now, I am actually joining you from my room, as you can see in my video. That's because it's very early morning, while it is evening for you guys. So I'm joining from a city called Montreal, that is in the eastern part of Quebec, eastern part of Canada. So as you can see in that pointer there in the global map. And this is our university, one of the university in Canada. And where I am joining is from the off campus. Our campus is a bit away from the main campus. And as you can see now, it is the time in Canada. You can see the change of colors of trees, and that's because it's the fall season. You can see trees that comes in different shades, and this is not Photoshop. This is the right time to go, and one of the best places to see the foliage, as we call, is Montreal or is Quebec. And so this is the time when it happens. So it's a beautiful campus. So I hope one day you will be able to come to Canada and visualize this magic of nature that goes through every year. And so in my presentation, I will be happy to introduce to you, talk about what is nanotechnology, what is nanomaterials first, and then we'll talk about some of the applications and also the other side. As you may know, all the different great technologies actually do have some side effects. So we will also look into the potential implications of nanotechnology applied in food and agriculture, and then now go to conclusion. And also I should say that the studies that I'm going to share with you are mostly from my own group. There are a different number of studies that are happening on these topics, but there are not many studies on nano, particularly going into food and agriculture application. All right, so let me start with what nanotechnology. So if you want, for you to learn what nanotechnology is, you want to understand what nanomaterials. So most often you might have heard about atoms and molecules that build up or they arrange in space to form materials, or that what we see as a matter. So in its bulk form, that's mostly what we know is atoms or molecules separately. What you see is their manifestation as bulk materials. So if you see as a gold, you see the golden colored gold bar or the ornaments that you wear. Or you see, for example, iron bars as bigger forms. But when you know that big forms of actually is made up of atoms or molecules in some instances. So when people talk about nanomaterials, I had heard people talk me are this than atom. So first of all, I should say that they are not. They actually have to say where it starts. I don't know what happened again. Okay, here we are. So a few of the atoms, there are a definite number of atoms, they come together. We call them as a nano clusters. So if you look at the size range, so the bulk material could be in the range of, let's say if it is a micron-sized particle that is 1 by or 1,000 times smaller than a millimeter, that's what we call a bulk material as a bulk material. Is from the bulk material, if you go down in the scale to nano, which is 10 to the power -9 of meter. So you can imagine that is almost a million times smaller than a millimeter in your scale. So that's where you will see a few number of atoms, maybe gold, that have 25 atoms come together. So that's called cluster. So it's called nano cluster. It can be made from metal, different kind of metals, for example, silver, gold, platinum, iron, and you name it. And also you can form metal oxides, titanium dioxide, zinc oxide. All these are examples of metal oxides. So similarly, you can also get functions with biological systems. Okay, so if you look into nature, what you will realize is at the very fundamental level, many of those principles or many of those functions are done by nanometer scale materials. So if you look into your cell, for example, there is an organelle called mitochondria, which is also known as the powerhouse of the cell, which is like a battery to operate the cells that constitute our body or tissues. Now that mitochondria have machineries, protein machineries that are operational at nanoscale. So what we mentioned about this is when you look into nature, the perfection of engineering, it can be seen at nanoscale. So that's why you can see things are working flawless. All right, so so I just want to make sure that you understand the concept of nano. Nanomaterial is not below atom. It is actually a collection of atoms arranged in specific of a nanocrystal, or it can be in without any, or as in the case of an amorphous. So I hope you understand the scale and the
ചെറിയ ടെക്നിക്കൽ എറർ ഉണ്ടായിട്ടുണ്ട്. സജി സാർ ഉടനെ തന്നെ ജോയിൻ ചെയ്യുന്നതായിരിക്കും. സജി സാറ് ലെഫ്റ്റായി പോയതാണ്, ഉടൻ തന്നെ ജോയിൻ ചെയ്യും. ദയവ് ചെയ്ത് അതുവരെ എല്ലാവരും വെയിറ്റ് ചെയ്യണമെന്ന് പ്രത്യേകം അഭ്യർത്ഥിക്കുകയാണ്.
Sorry, I didn't know that I got signed off. Can you hear me now?
Yes, sir. Yes.
Now, okay.
Okay, so I don't know where we got cut off. I was talking about the nanoscale and the arrangement of atoms to make the
It was about nano clusters.
Okay, good. We'll start the exercise all over again. Let's see if it will. Can you let me know when you can see my screen, the present slide?
Okay, Saji. Now it's not seen. I will let you know.
Okay.
Now it's coming, Saji.
Okay, good. So what I will do is I will also join from my phone, so I know that. Okay, good. So I was talking about how the carbon atoms come together to form the carbon, the icon of nanotechnology or nanomaterials, which is the carbon 60. It's also known as keyboard. So the atoms are specifically as atoms come together to make, in this case, a molecule called carbon 60 atoms of carbon. So you can see how they are arranged in space to form this molecule or nanomaterial called carbon 60. And there are other forms of carbon nanomaterials. For
Sarana. Saji, now the voice is not clear, but you can see the slides. Saji, now it's not audible, but we can see the slides.
hello, can you hear me now?
Yeah, now it's clear. Now it's clear.
Can you hear me now? Can you hear me now?
Yes.
Can you hear me now?
Yes, sir. Yes, sir. Yes, sir.
Can you hear me now?
Yeah.
Yes, sir.
Yes, sir. Can you hear me now?
Yeah.
Yes, sir.
Yes, sir. Can you hear me now?
Yeah.
Yes, sir. Yes, sir.
Okay. So in that case, I will I'll continue my presentation. So let me go back again. Let me know if you can see my my slides as well. Okay, so we don't. I will try to speed up. So the discussion is about what makes nanomaterials unique. So if you look into the hierarchy in the structure of atom, in the hierarchy in the structure of matter, you can see it starts from atoms or molecules. They arrange in specific manner to form bulk materials, materials that we often see. So most of our understanding about matter comes from either as a bulk material. So for example, gold we know as a gold bars or ornaments, and also we know gold atoms or molecules. But there is a space in between, and that is where the nanoparticles comes in. So one thing you need to understand is when they are in the bulk state, the properties that govern are according to Newtonian physics. For example, their movement, their interaction with light, etc., are governed by Newtonian physics. But when they are in the when they are in the atomic scale, or when they are an atom, the Newtonian physics does not apply anymore. It's mostly about the quantum physics, and that's because there is a definite energy band and etc. that governs their interactions, their movement, their interactions with light, etc. So, but there is a state in between. When the nanoparticle actually sit in the state in between, partially they obey the Newtonian physics, partially they obey the quantum physics. So there is a dominance of quantum effect, and as a result, you can see discrete functionalities, as in the case of colors of gold particle. When you change, you can tune the color of gold by tuning the sizes in the nano range, for example. That's because of a dominance of quantum effect. But most importantly, one thing you need to understand is when the materials are in nanoscale, their surface area increases tremendously. And that's because they have more surfaces exposed because they are smaller, and so the surfaces are exposed. And that's also another phenomenon that happens is the surface exposed atoms. So those which are labeled in red are the atoms or molecules in a nanomaterial that is exposed to the outside world. So external environment. So there is a tremendous increase in the number of atoms or molecules that are exposed to the outside world. And then there is extremely smooth surfaces, so almost defect-free. There are reasons, I mean, I don't think you need to go in deep to understand all, but you need to understand the surface area increases, more atoms or molecules are present on the surface. As a result, they may have unique properties. And here is an example of how the surface area increases if you divide or if you break down particles into smaller sizes. Starting from a 1 meter cube particle, the surface area is around 6 centimeters square. But if you have the same volume of the material, but if it is divided into smaller cubes of 1 millimeter, now the surface area is 60 meters square. But what happens if it is the same material, same volume, but you just divide these into smaller particles of 1 nanometer scale? The surface area is 60 million centimeters square. So you can see with the same amount of material, now you can cover this much surface area. For example, it covers more than one football stadium with a 1 centimeter cube of material. So that is the power, or exemplifies how the surface area goes or increases as the size decreases. So here is a demonstration or just pictorial representation of what do you mean by the surface exposed atoms. As in the case of this particle here, the golden colored balls indicates those that are present on the surface, and the blue that are in the inside of the particle. Now, if there is any reaction to happen or surface phenomena to happen,
it is mediated through the atoms that are present on the surface. The atoms that are present in the core of the particle does not interact with the environment. And so when you divide this bigger particle into smaller particle, you can see the percentage or the proportion of the atoms that are exposed to the outside is increasing and tremendously. So as a result, there is an increase in the surface exposed atoms. So if you want to see what happens if this phenomena dominates, you can take the example of diamond and gold, because these are some of the materials that are precious. And you know about some of those characters of these materials. As we know, diamond is the hardest natural material known to man. Now, if you try to bend it, you cannot imagine about bending it or this material behaving like an elastic material. All right, so it's brittle. So if you apply a higher force, it will not bend, but instead it will break. And you will end up in two pieces of the material. So, but what happens when it is in the nanoscale? What you're looking at, this cone-like thing here, is the cones of a diamond that is deposited on a surface. And then there is an intender that gives a force. So you can see the cones, as you can see here, when it is in contact with the intender that is applying a force onto that, it bends like this. So it's like it is behaving like an elastic material, which is unknown otherwise. So you can see when this material comes in the nano size range, it changes its behavior from a hard, brittle material. Now it behaves like a rubbery, elastic material. Imagine about that. That was something that we could have never thought that this material will behave like. Now, that's because the surface is extremely smooth. There is no defect here for it to crack. And so it can go up to, for example, 10% of bending. That is something unknown, previously unknown. So I was talking about gold. The gold, as we know, as the golden noble material. Now, when you reduce the size, you can get a spectrum of color, depending upon the sizes in the nano range, not in the microscopic range, but in the nanoscopic range. So you can see it starts from extremely small. It gives something like a cherry red. When the size increases, it can be like in a bluish range. So that happens because of the quantum dominance, the way it interacts with the light at the nanoscale. So there are many other properties. Because of the interest of time, I won't go through different properties of nanomaterials. All what you need to know is when the material size is reduced to nanoscale, we begin to see some novel properties, properties that were previously unknown. And there are empty number of applications of these novel properties. Now, what is that application? That is the nanotechnology. Manipulation of matter at the nanoscale to generate size-dependent functionalities, different from atoms or molecules or bulk materials. So the reason I gave this definition is, you can see when you manipulate materials in the nanoscale, their properties change. So they attain novel properties, new properties, and these new properties can be used for different applications. So I saw that gold nanoparticle, for example, it is a noble inert material. That's why you wear it, you do business with it. If it was to get rusted, you will not keep that noble material as an asset, for example. So, but when the material gets in the nano size, gold, as we know, the noble material is no more noble. It is reactive. It can react with other materials. It can react with other molecules. And the best example is the one which we know as biologically inert material is no more biologically inert. It can be used, for example, cancer treatment, because we can use it to kill cancer cells, because the size, if you reduce it to something like in the nanometer range, for example, 3 or 4 nanometer, it can get intercalated between the major grooves in DNA that makes our nucleic, the nucleic acid that makes our nucleus that are present in the nucleus. And when they intercalate in the major grooves of DNA, that cells can be pushed to apoptosis or a death. So, and in the case, you can use it for treating cancer, and it is being used now. And that's an example of how the material property changed and that novel functionality is made into an application. As in the case of carbon nanotube, we saw it's 100 times stronger than steel, but only 1/6 the weight of steel. Now the application is we can make it for building up the bodies of aeroplane. So you get lighter aircraft, but with the stronger bodies. And you will see many of those carbon reinforced sports goods, especially as rackets for badminton or tennis rackets you might have seen, and also for example bicycles that are lighter, and that's because it is reinforced with the carbon nanotubes. And so that there are, these are some of the examples of exciting applications, and applications can be in different sectors. So for example, as in the case in our recent times, we are looking for cleaner technology. So we can realize many of those clean technologies. For example, clean energy. It need not be very polluting. You can use nanomaterial-based solar cells. Nowadays, there is a new form of solar cell that is going to come to market, which is enabled through nanotechnology. Clean water, filtration of water to make clean water available to everyone without the cost of a filtration techniques that are available at the moment. Clean air. That is because we know the air pollution is a major issue. So if you want to breathe clean air, you need to filter. Similar to water, you need filtration systems. So nanotechnology is enabling filtration as well as capture of carbon monoxides or carbon dioxides to convert it into fuel or other kind of molecules. Clean land, to clean up the lands that have been dominated with, for example, pesticides or those kind of toxic chemicals, and of course for good health. But you can see when you get time, probably I will share this slide. You can go through these different applications which are available as of now. This is not a technology that we are talking of the future, but a technology that we are talking about what is happening right now. So in the case of application in food and agriculture, what is good about nanotechnology? So I will give you some examples how nanotechnology is enabling applications in food and agriculture. So we know world population is growing. So by 1950, according to UN estimate, world will be populated with 9 billion people. So currently it is 7 billion. So hopefully coronavirus will not change that statistics too much, but still you will definitely, whatever there, even if there is corona, we will see a great increase in the population. So what it means is for the feeding of the population, extra 60% of food that is required to feed the expanding world population. And as you may know, as of now, around 10% of the world population experience what we call as a severe food insecurity. So that is one scenario. The question is, can we actually increase the production? Answer is yes, we can increase the production. But another question is it sustainable? So we may be able to feed the next generation by increasing the production of agriculture producers as the case is done now. But if you continue in the same trajectory, we may not have food or land or water to feed the generation afterwards. So the question about sustainability is important. And another issue is the safety of food that we consume. There is, as you may learn in your food science, there is food safety issues that comes from mostly from microorganisms. There's a lot of people affected by the contamination of food with pathogenic microorganisms that cause diseases. And the problem is mostly underestimated because many of the cases go unnoticed. So 1 in 3 of children below 5 years old die from foodborne diseases. That is a scary statistics, but want to know that there is lots of issues with the food safety. So World Economic Forum has put up different strategies or technologies, what you call as transformative 12 technologies to enable production of agriculture producers in a sustainable manner. So that is, there are call for alternate protein. So nowadays you might have heard about plant proteins being replacing or plant proteins replacing animal products. Nowadays, when you go for shopping in at least in this part of the world, you can see vegetable patties or burgers made of vegetables protein, not with any animal protein. So alternate proteins, there is a demand for alternate protein, cleaner proteins, because it takes a lot of resources to make meat, but instead if you can get the same kind of texture as a meaty texture from plant proteins, it will be preferred. Then there is sensing and tracing of producers. Now we have no idea about from where the food is coming from, because now the food business is so complicated and it has gone beyond boundaries. Countries are trading, and the trading of food commodity is a major business, and as a result, you will be receiving food that comes from different parts of the world. In the particular case of India, for example, or Kerala, for example, you may not experience many of the food coming out from outside countries, but in the case of countries like Westerners countries, most of the food food travel across the boundaries. But if you want to know where it originated from, for example, if you're talking about berries, cranberry in Canada, often we are looking for cranberries that are grown in Canada. But unfortunately, there are reports nowadays surfacing that these kind of berries some of the berries are grown in China in a more uncleaner manner, and then they are going to, for example, Chile, and then there they get stamped as organic. I think it was the case of cranberry. One of the berries here, and it get exported to Canada. So see the route from China, it originated, and it reached Chile, and it is exported to Canada. And the Canadian market, it is sold as a Chilean berries, but and organic. Or when it is no more, it is not at all organic, and it is related to many of the rotavirus infections or outbreaks. So that is how things are happening nowadays, the business. So now we need more smart tracing technologies. Food traceability as a subject is actually getting more attention these days. And one of the subjects that I teach is also food traceability. So there are smart methods. We need smarter methods like blockchain enabled food traceability, for example. And also there are lots of requirement of data. For example, you might not have heard, there is something called as a smart agriculture, precision agriculture. What it means is you have sensors embedded in soil and in plants, and you get real-time data from the soil and the crop plants. And so you can real-time monitor whether the plants are getting enough nutrients, enough water. And those data are collected by, for example, a satellite placed in the orbital, and that satellite, depending upon the taking the message from the data, it can instruct for the addition of water or fertilizer to the land, all done automated. So that is precision agriculture. So you don't have to spend more fertilizer, more water. You only spend that much what is required to raise that crop, and the optimal amount to get the best produce or the best cultivation without spoiling environment. Okay, so and also there are other scenarios where there is a accumulation of data. So these are some of the scenarios. Why I'm explaining is, another reason why I'm explaining is because in future you may deal with many of these technologies or these newer things happening. Okay, so you may be coming across positions that demands more of a data handling, or what you call as a big data handling, etc. And then there is something called as Internet of Things. It may be also a new terminology for many of you. So nowadays, sitting at home, you can watch this video, for example. So the next level of this thing is controlling your devices remotely. For example, if there is a person sitting as an administrator for this meeting here, he controlling my voice or my microphone as well as my video. So right now he can switch on and switch off my video, but imagine whether he can actually move my camera around to see where I'm sitting. Okay, so that is possible. Nowadays, there are technologies. Even now, you might have seen that in your places. There are cameras that you can fit in your houses, and we can be working remotely, and you can looking remotely. You can actually move the camera. That is because of enabled through a technology called as Internet of Things. And that's also happening in the case of food production. For example, if you're harvesting fish from sea and you want to know whether that fish is harvested in a more sustainable way, more of a earthly, sustainable manner. So you can have technologies enabled through Internet of Things. You can have real-time monitoring where the fish is being captured, where it is being placed, what is the temperature in which it is kept, if the temperature abuse is happening, etc., can be monitored remotely. So you are actually at a juncture of time in time when these kind of new disruptive technologies are going to take over many of the traditional or conventional things. So many of the things that you will learn will become very fundamental, but you may want to get ideas about all these things in order for you to go into a competitive world that is waiting for you. So there are many other things. Another thing is gene editing that is happening through the another enabling tool that was recently discovered, and the recent what's that called? Nobel Prize for physiology for medicine was, in chemistry was for CRISPR-Cas technology. That is a new gene editing tool that enables anyone. You can even edit the genes for crops or for animals. Nowadays, there are animals, for example, cattle born without the horn, that is made possible through CRISPR-Cas, CRISPR-based genome editing of cattle, for example. And you have recently there was this salmon that was approved by FDA, which is also a gene-edited version of salmon that grows even in the colder climate. So these are some of those exciting applications, new tools that are coming into food and agriculture. So many of these exciting applications or the transformative technologies can be enabled through nanotechnology. When we talk about Internet of Things, when we talk about precision medicine, or CRISPR-Cas or CRISPR-based genome editing, you can introduce those kind of machineries or that RNA sequences into the cells of plants or animals using nanotechnology, and also different methods of alternate proteins, even lab-grown meat. There is an exciting technology that is coming. Now we are getting meat from animals. Imagine about the possibility of growing meat inside the lab, and that's happening in many of our labs. Like we grow the cells, and there is one more layer on growing it as a tissue for obtaining meat. All this, what I meant is nanotechnology can be applied for different sectors. So when you look into the nanotechnology applications in food and agriculture, right from the food production stage in the agriculture, for example, as a fertilizer, pesticide, drug delivery, infection control, etc., for both animal and plant products can happen, can be enabled through nanotechnology. And when it comes to food processing, then there are food processing surfaces that can be made super hydrophobic or super hydrophilic. So that is important for food, for example, to prevent the accumulation of bacteria and biofilm formation, to get much cleaner surfaces, and also to get containers, food containers that can decant very well. For example, containers of tomato ketchup. Nowadays, you can see not even a spot of tomato will be adhering to the container because the surface is modified. And flavor enhancement, colors and flavors are enabled through nanotechnology, and tracking and sensing, and also more intelligent packaging materials, new packaging materials that tells you whether the food is wholesome and good to consume. It can detect and tell you if there is a presence of pathogenic microorganisms. All these are exciting applications that are now being applied, and it is almost $1 trillion business. So I just explained one technology that was recently published in a journal, which is about introducing nano as a fertilizer. So here is a group that synthesized or generated a nanomaterial based on based on hydroxyapatite and urea as a coating onto the hydroxyapatite. This nanomaterial is made from materials that make up the bone. That is how this material will look like. So if we apply it into a field or water, you can see the release of urea in the case of this nanomaterial is slow. And slow and sustained release of urea is most important, because if you put urea as urea, immediately dissipate as urea, and as a result, the plants may not be able to absorb the nutrient. what happens is there is more of pollutions. You might have heard about the algal bloom that happens because of the fertilizer going into over fertilization of the land. But if it is a sustained release, you can see for the half of the amount of fertilizer, you can get production higher than what you usually get with the urea alone. So this is an example where fertilizer can be used, can be made to a nano form to increase the production or efficient use of fertilizers. Here is an example from my own study. Prior to my appointment in McGill University in Canada, I was working in Singapore for a while. One of the exciting projects that we undertook was about aquaculture. That is about growing fish. The fish that you see is called sea bass. I'm not sure what is the Malayalam name for it, but that is a very tasty fish, and it grows really big. In a year, you can get something like 1 kilogram fish. If you go, for example, 2 years, you get something like 3 or 4 kilograms of this fish. But the problem with that fish is, or with any aquaculture for that matter, is if there is any infection in an aquaculture farm, you will destroy the farm. That's because the fish are grown or cultivated in high density. They are together high density. So as a result, if one of them get infected, it will spread and the entire farm will be gone. Almost 90% of the fish will be dead. So there is a need to control the infection. So one of the technologies that they use is inject the fish with a vaccine. That is, you isolate the bacteria that causes the disease, make vaccine out of them. Now, if you imagine if you have 1 million fish in a fish farm, you need to knock out all the fish, and then you have to inject the vaccine as you might have gotten the vaccine yourself. Now, the problem with that is you need labor skill, people to inject it, and also sometimes you get self-injected. So those are some of the issues. It's a highly expensive process. So what we develop is a nano vaccine. What it means is, instead of the syringe in the microscopic level, you made a nano syringe, a syringe at a very nano scale, which can actually accommodate the vaccine component, that is the outer membrane protein of the bacteria that causes the disease. So when the fish is kept in a water that is suspended with this nano vaccine, they get self-injected. So when they breathe, their gills they take up that vaccine through their gills or through their mouth, and as a result, the fish get immunized. So you don't have to do this kind of injection, and also you can treat the fish at a much earlier stage, which otherwise require the fish to grow to a certain length before it can be injected with the vaccine, as in the traditional way. So this technology is now applied in fish in a farm. So other applications of nanotechnology that involves, again, research from my own group and from many others have shown that nano can be used for introduction of nutrients, especially micronutrients to people. So that's, for example, in the case of anemic people, you can introduce iron as a nano iron, so it enhances the uptake of the iron, otherwise the body may not. And calcium, for example, if you put nano calcium, there are milk products nowadays available in the market that comes with this claim of nano calcium that is in the nano form, which will be absorbed by the bones, and so you get stronger bones and stronger body. And that is especially important in the case of women health. So another kind of application is silica particles. So you might it may look foreign to you, but you have tasted this silica particle only without your knowledge. So why is the silicon dioxide particle added in food? By the way, what is silicon dioxide? It is nothing other than, if you know the fine sands. So the chemistry of sand is also silicon dioxide. So you can chemically synthesize pure sands. So earlier, people used to adulterate rice or pulses with sand, and that was in the very high microscopic level. And that's because and when you bite onto that, you lose a part of your teeth. Okay, nowadays it is not in that microscopic level, it is in the nanoscopic level. You bite into it, you will not notice it because it is smaller than the grooves on your teeth. Now, why is it used? Is silicon dioxide is used in powders. For example, as you can see in this case here, is a spice powder that is kept with and without the silica particle. The one without the silica particle get clumped and it won't flow nicely, while the other one with the silica particle will flow nicely. And that's because the silica particle absorb the moisture in this powder. So to keep the food powder separate well enough, so that they've increased the flow nature. So it is called anti-caking. So when it is clumping in the presence of moisture, it's called caking. So when you add silicon dioxide, it's an anti-caking agent. So it can be added to many of the spices, salt, sugar, you name it. It's a high volume production material. And if you are consuming a lot of these kind of products, not the one spices that you make at home, if you make your own powder, but if you are buying from outside, you can see there will be a lot of these kind of spices. And nowadays, even the table salt is not really clumpy as it used to be earlier. That's because it is added with silicon dioxide. So much so that you are exposed to this kind of chemical silicon dioxide almost 100 milligram per day, depending upon your body weight, and that's because of the consumption of this kind of food. Okay, 100 milligram per day is not a small amount for a material like this. Another material is titanium dioxide. You might have heard about titanium again in the case of sports goods or your dental materials, but titanium dioxide is such a versatile material that can be applied in food as well as in your creams or facials, etc., and also in your hygiene products like dental or toothpastes, for example. What is the application of titanium dioxide? It is applied as a white pigment. That is the best pigment known to human because it has such a high refractive index and masking power. It can be applied. So many of those chocolate items or candies contain heavy amount of titanium dioxide applied because that provides a good color or a pigment. And as a result, you can see people especially kids, are getting exposed to titanium dioxide at much higher rate. Something like milligram per kilogram is shown here. It's almost around 200 or 100 to 200 milligram per day. And one of the studies that we did is, if you are familiar with this chocolate called M&M, so you can see, you might have seen these beads coming in different colors. These beads are from shades of yellow to blue to red to green, etc., etc. So we estimated the titanium content in this product, and we, to our surprise, noticed that the titanium is higher in the light shaded material, and that is reason is because you need more masking power, and as a result, you can see if you bite through that the beads of this chocolate, you might have seen layers. The first layer is actually a mix of sugar and titanium dioxide that provides the hard shell for this product. All right, so those are some of those technologies that are enabled by nanotechnology. That is only a few examples. There is a lot of examples of nanotechnology going in agriculture and food, and there is it is used for increased food production, enhancing food quality attributes, enhancing nutrient absorption, improved food safety, like warding off against microorganisms, and improved shelf life of products, like you can store the products for a longer time. So those are some of those applications or some of those functions that are realized through use of nanotechnology. So you may see that this is all benefit. What is the bad side? What is the adverse effect of nanotechnology? I'll give you some example based on my own studies, but you have to understand there's a lot of other kind of studies done in that regard from my previous work as well, to understand what is the contribution of the material size, because you know this is extremely small size. When the size is small, our body or the biological system is built to deal with either molecules or micro or macroscopic substances. Our body is not built to deal with engineered nanomaterials. As a result, these materials can creep through different barriers on our body to reach into remote areas, like even if you consume, for example, silicon dioxide, it can reach up to your brain. That's because they can go through different biological barriers. So I'll give you one example of a study that was done from our group. So when you consume products containing nano, it goes through different places. First, the journey starts from mouth, it goes to stomach, small intestine, and large intestine. So it can encounter different environment. So, and in different environment has different kind of biomolecules and salts. So it starts with saliva. So we wanted to see what will happen if nanomaterial interact with saliva. There are studies done a priori. We are to see how the surface of nanomaterial absorb molecules, especially proteins. And so there is a concept called, in this case, a depiction of a core particle and the particle surface absorbing proteins or biomolecules. It can be proteins, lipids, carbohydrates, etc., depending upon the surface chemistry, the chemistry of those surface exposed atoms that I explained before. What you need to know is there is different layers of proteins as well as biomolecules that are formed when a nanoparticle enter human body, when they interact with the human biological fluid like saliva or human blood serum. Okay, what is the implication of that? So first we studied different materials that are used in food products. For example, we studied silicon dioxide in the nano form as well as in the microscopic level, and titanium dioxide and silver, that's another nanomaterial that is coming in contact with food. And we interacted with human saliva and we looked for that proteins using advanced techniques, and then we'll not go into the details. All I want to tell you is there is an enrichment of protein. The proteins that have vital function in host defense. In saliva, there are some proteins and peptides that ward off against the microorganisms. If they are not present in that saliva, what will happen is the microorganism bacteria that goes into your mouth will actually invade your other cells or your teeth, and then you get all these issues with your teeth decay or other bacteria infecting your mouth. So that is one of the functions to ward off against microbial pathogens. And then there are other enzymes or proteins that do have enzymatic activity, that they act as they break down bigger molecules of starch. For example, the example is amylase. That's an enzyme that actually work on the starch to divide. So what we see is there is a preferential adsorption, i.e. what it means is there is no even though the protein is present at a very low concentration in saliva, you can see them at a much higher concentration when you retrieve the particle. And that's because there's a preferential absorption of different types of proteins. And those type of proteins are mostly to do with the host defense and enzymes. So what happens is that when the protein absorbs, for example, this is salivary amylase, the enzyme that catalyzes the digestion of starch to the maltose or smaller units of starch for its further degradation, further digestion in the intestine to release glucose that provides energy for our body. So what happens when it interacts with a nanoparticle? What we saw is the salivary amylase is inhibited, because the function, when it is adsorbed onto the surface of silica nanoparticle, for example, it's not happening with micron-sized particle or titanium, but with the nanoparticle, they actually have reduced the function of that enzyme, which means it will not be digest that starch, or it's partially inhibited, not fully, partially inhibited. For example, around 35% of inhibition at a concentration of silica that is likely to encounter in saliva during consumption of food products that contain silica particle. So that's because the structure changes, and we have seen that the structure of the enzyme is altered. So you may also learn in your degree, when you are learning, that the function of a protein is very much dependent upon the structure. Once the secondary or tertiary structure is altered because of chemicals or because of interaction with nanomaterial, for example, in this case, you will see their function is compromised. So similar example with another enzyme, that is trypsin, involved in the digestion of protein. What we saw is, again, nanoparticles of silica are more prominent in inactivating or in inactivating or interfering with the function of a trypsin. So I will not go detail. Again, the function is compromised because of structure. You have tools to study the structure of protein. For example, circular dichorism or FTIR. These are some of those tools that can be used to see the structure changes. And you can see when the nanoparticles interact with these enzymes, the structure of the enzyme change. As a result, it compromises the function. And you can observe that compromising function as altered protein splicing. The protein, if it is not spliced properly, if it is not cut into its units, the body may not assimilate the nutrients. So what it means is nanomaterials, from our own study, shows that interact with proteins in human body, and their interaction can cause the changes in the structure, and that change in the structure of the protein or enzymes in this case can alter the function. And so the study suggests that nutrient assimilation may be affected when you have lots of nanoparticles in your diet. Another example is what will happen if this reaches our gut and the small intestine. So you know that I mentioned to you, our body is built to withstand the invasion by foreign materials, either as microorganism or as virus or as even particles entering our body, because we consume a lot of particles. And usually I said, like, our body is built to withstand or to prevent or ward off from the molecules. So there is a selective absorption. When things reach the gut and the intestine, not everything is absorbed by the body. There is a selective absorption. Body absorbs only what it needs. Okay, so there are receptors, there are transporters for nutrient absorption. There are mechanisms that ensure that only things that are needed for the body majorly are absorbed. But there are things that can creep through. But in the case of nano, so what we can look at is the small intestine. Intestinal wall is actually covered with a layer of cells called epithelium. And they have to be close together. They have to be tight. So there are some machineries that make sure that the cells are held together tightly. They're called tight junction. This is protein that actually binds together these cells, so that forms a tight junction. So there is no gap here for things to get through this tight junction in the epithelium. Because below the epithelium is the fibroblast, and then there is blood vessels. So if things get passed through this layer, it can reach the blood. When it reaches the blood, you know that it can reach anywhere in the body. And there are machineries for that. You can see the tight junction protein, and then proteins inside the cell that actually make sure the cell size and shape is also maintained. So there is that integrity of these machineries here, the protein machineries is really important. That's what you need to know. Okay, so what happens when these kind of particles interact with epithelium? We studied that using we can make this kind of epithelial layer in your labs. That's why I said, like, you can grow the cells. This is like a human intestinal epithelial layer. You can see there is we call as a transwell assay, where there is a layer where the cell is growing, and then there is a basolateral layer when there is the below this the line of the cells. So you can cultivate that cell, and you can put your things here, for example nanoparticles here in that apical chamber, and see if things are actually going through that to see it outside, or you can even see whether that layer is disrupted through staining, through microscopic examination. So when we look into that, what we find is some of the particles are good to cause that kind of disruption of the cell junction, and you can measure that electrically. There is a way of measuring the conductance of this layer. So if this layer is maintained, the electrical the resistance is higher. When there is a seepage or a leakage, you will see that resistance going down. So first we see there is an increase in the oxidative stress that is caused by the exposure of the cells to these nanomaterials. And as a result, you can see there are pro-inflammatory cytokines. I won't go in detail about that, but they are the mediators that are produced by the cells that causes changes in this permeation. And you can see the control cells in this case is a confocal microscopic image of the cells. You can see they are held together tightly, but you can see the junction between the cells increases as a result of its interaction with the nanomaterial, basically telling you that the epithelium became more leaky when there is an interaction with particles of nano that is used in the food products. And you can also measure the resistance, and you can see with some particles like silver as well as with the titanium dioxide, that epithelial resistance is decreasing as a function of an increasing time or concentration, and that's because they have an effect on tight junction. So we have done more studies to see what happens, and we know that it is because of these particles interacting or causing the inflammation, and the inflammation causes disruption as the tight junction. As a result, you get a leaky memory. So here is a depiction. So when you have the particles, they come in contact with the membrane of the epithelial layer. It causes disruption of the tight junctions as a result of the inflammation that happens. So to say that there are some unseen or unexpected behavior that can be exhibited by nanomaterials that are likely to enter our diet, or that we are likely to consume. And another example is silver. I said silver is one of the material, high volume production nanomaterial that is used in consumer products and food packaging. The main reason why it is used is to get rid of microorganisms that causes spoilage of food, or pathogenic microorganism. And nowadays there is something, the clothes called odorless socks or odorless underwears, that is enabled through the nanotechnology. That's because this fiber in the textiles is embedded with the silver particles. As a result, the microorganism may not be growing. Microorganism can't grow. If the microorganisms cannot grow there, they cannot make a foul smell. And that's why you will get something called as odor-free clothing. Nowadays, it is available especially for sports goods and socks. And another formulation that is made is emulsions of nano silver that is used for agricultural application, especially in animal and poultry farms, to get rid of microorganisms. And from our own study, we have seen the possibility we have developed the technology to functionalize, for example, a wound dressing gauze with a silver nanoparticle that has shown excellent activity against bacteria. But I will give you an example where this goes in the wrong direction. We examined one of those food packaging material that we tried from in shops in Singapore, which claimed to have silver embedded in this plastic material. In fact, when you analyze that material for its presence of silver using technology called ICP-MS, or atomic absorption spectroscopy, you can actually look for metals. In this case, we use something called as ICP-OES. We saw that silver is present in these materials. And we took 3 samples of this plastic. 1 sample does not have silver, 2 others have silver. And we find that if you keep that milk, for example, as an example of a food product store in this, there is almost 12% of that silver get migrated to the milk. What happens is, when you have that small amount of silver that get translocated or transferred to the food that is stored, instead of preventing the microbial growth, it actually enhances the microbial growth. So you can see here is a CFU that is stands for colony forming units. You might have done the microbiology experiment to count the bacteria from food products, for example. And you might have seen how to count and extrapolate. So what we are seeing here is the CFU, that is colony forming units, counted from the milk that is stored for different days. And the 10th day, you can see these 2 bars are higher than this bar. That's because these 2 bars represent sample 2 and 3, which has silver, and this one represent which does not have silver. For example, an acceptable level of microorganisms or total plate count is something at 5 log, CFU 5 log. And so you can see for in this particular case, on 10 days at 24 degrees Celsius, you can see the sample which does not have silver is below that level, microorganisms is below that level, but with that silver, the microorganism level is higher. And that's because there is a phenomenon called Hormetic Effect. What it means is, when bacteria exposed to small amount of any kind of bactericidal agent, like it can be antibacterial agents that are used in your hygiene products, in your soap, in your dental products, or in this case, silver. when it is in extremely small concentrations, the bacteria actually resist it, and as a result, there is an increase in the metabolic activity of the bacteria, and they will divide faster. So there is, just before the killing starts, the concentration, there is a concentration or range of concentration that increases the bacterial number. So you can see it is actually having an opposite effect. It is not like, in some cases there is no effect, but in this case particularly, we can see there is a opposite effect. The silver is applied in food packaging to prevent the bacterial growth. Now you can see that because it is extremely small concentration, it is enhancing the bacterial growth. So these are some of the examples of bad effects of nanotechnology. This is only a very small shot of examples. There are empty number of examples where the nanomaterials can go and do with our human cells. For example, titanium dioxide has even shown to be causing malignancies, and as a result now titanium dioxide is banned from food in France, but I believe is not banned. I mean, I don't think there is any ban of these kind of materials in India or in other parts of the world. And silicon dioxide, as you see, is used in many kind of food products that you consume, especially for products that are ready to eat. And that's because the manufacturing process is facilitated when there is a silicon dioxide. And so to summarize this section, so nanomaterials in food can adversely affect the function of digestive enzymes. We saw that example. Cause epithelial leakage. So you can see that they don't respect the biological boundaries. They can seepage through the biological boundaries and can enter tissues that are not intended for them, and cause adverse outcomes such as inflammation. There are studies showing, and from our own group has shown, that nanomaterials can cause inflammation, chronic inflammation that underlies many of the disease conditions, including cancer, and then pre-neoplastic lesions, for example, nanotitanium dioxide causing the pre-stage of cancer that has been proven in animals. So there are some of the adverse side effects of nanotechnology going into food. So we saw the benefit of it in different aspects with respect to food production, food processing, with flavor enhancement, nutrient absorption, etc., etc. But here is an adverse effect showing that nanomaterials in food may also have the other side. Now, how can we change? The question is, can we change the outcome? Or if so, how can we change the outcome? So the answer is yes, we can change the outcome. So if you want to make it more towards a beneficial side, that is in our hands, how we understand the mechanism of its toxicity. From my group, we have previously shown that zinc oxide is another material which causes toxicity because it dissolves and releases toxic zinc ions. So if you actually prevent that dissolution through a process of introducing iron into the crystal lattice of zinc oxide, you can prevent that dissolution. As a result, the material becomes safer. So there are many examples of making or re-engineering of nanomaterial to make sure that their toxic properties are taken care of so that we enjoy the beneficial side of it. So the answer is yes, we can change the outcome if we know the forces that shape nanobio interactions. So before I conclude this lecture here, I will give you a philosophy that you should keep in mind, because when you grow up, you will be in places where you have to gauge a technology, you have to develop a technology. So you may be involved in that kind of processes. So you are the next generation leaders who will be involved in those kind of processes. So you may have to see, what can I model if I want to develop a new technology or a new method that goes into food? Especially in the case of food, I believe we do have a commitment for public health and safety. So you do have certain level of responsibilities as a food scientist, or as your role in a regulatory agency, or your role as a quality control person in a food industry. You do have certain level of responsibilities, and that responsibility demands you to know, what can I model? So for my philosophy is simple. So if you are looking at a technology, the technology should be simple. It should be economically and environmentally sustainable. Okay, that means it has to be viable economically. If it does not make any gain, financial gain, you are not going to introduce that technology if you are an entrepreneur. Okay, so it has to be economically and environmentally sustainable, which means it should not be toxic. For example, we introduced plastics. Plastic was a wonder material, but nowadays we are dealing with a phenomenon called as microplastics and nanoplastics. So if you take sample of water from the river that is flowing near to our college, for example, the Meenachilar, you will see presence of nanoparticles. Even in that kind of remote areas, you will see remote. I say is because we haven't, we assume that we haven't polluted those kind of rivers, but still you will be seeing what you call as a nanoplastics or microplastic in our soil, in our food, in our water. And it's unavoidable now. You cannot get rid of those kind of materials now. So it's an example where a technology was introduced. It's a fantastic technology. Plastic went into everything from food containers, food packaging materials, food processing units, and then in water bottles, etc., etc., etc. And as a result, now it is showing up in your food. So you will come across that kind of a phenomenon. You will see there is a lot of issues that we will be dealing with. So if you are a food packaging industry, or if you are a food industry, you should be careful about the type of packaging material that is used. So you should be asking this question: is it simple? Is it economically viable? Can I make this? Nowadays, there are people coming up with the straws that are made up of coconut. I think the palm leaf, or with many other kind of natural identifiable materials. But the question is it economically viable? Or can you make it at a rate you can make money? Okay, and also environmentally sustainable. Socially acceptable. Will people buy it if you introduce a technology? If you make the straws out of a palm leaf, are people going to buy it? If people are not going to buy it, again, that technology will not be viable. So these are some constraints: simple, economically and environmentally sustainable, socially acceptable, and most importantly, with an inbuilt design to anticipate and erase potential risk. That's where it gets really difficult. You have to anticipate what is the kind of potential adverse outcome that is going to, and that we are going to encounter once this technology has gone beyond, once it is mass produced and gone beyond your capability. Is there an inbuilt system for AIDS to be anticipate, to erase any potential risk? If there is any risk, can we actually mitigate that risk through an inbuilt mechanism? So, is there any technology that is that you are aware of? Usually, people answer me, there is no such technology. But let me tell you, there is a technology that you can actually model. That is simple philosophy. Is the pencil. You look at that pencil. Now, this is simple, economically and environmentally sustainable, and socially acceptable. Of course, this has a powerful tool. But most importantly, you look at this thing here. There is an eraser, and that eraser is built into this design to anticipate an error that could happen because of the application of this tool. So, when you want to imagine about a technology, this is the philosophy you should be keeping in mind. Is it pencil perfect? Pencil is a perfect example of a sustainable technology. So you should be asking, is it pencil perfect? So that's what we keep as our ideal material or ideal technology when we design nanotechnology for food and applications, because we do work on developing applications like you saw in the case of vaccine development, in the case for immunization of fish, or we do have technology that goes into plants for developing or enhancing the growth, and also other kind of materials that are being applied for infection control, etc., etc. So with that philosophy, I will conclude my presentation. I would like to thank my students who are the workhorses. They are the engine that materializes these kind of studies done. And of course, there is research granting grants that is required to do this kind of research. So funding bodies I would like to acknowledge. And more importantly, once again, I would like to thank the organizers, people who work behind it, especially Minimis. And also, I'm really excited and happy to see Alapat sir attending my talk. I never knew that I will have an opportunity to talk to him about my research or about what I do. And with that, I will be happy to take any questions from you. Thank you. Thank you, one and all.
Thank you, sir. Thank you for your valuable words.
Okay.
And this is
I hope
now
you are listening to me.
there is a discussion section. If the and there is a discussion session. The participants can ask questions in your dialog box, in your chat box. You can ask questions. Sir, there is one question which is one of the participants asked. Is the safety ensuring mechanism is proven or not about the nanotechnology, sir? Hello, sir. Hello.
Sorry, I can't hear
Hello,
you.
sir. Can you hear us?
Give me a minute. Let me fix what's going on here.
Okay.
Okay.
Okay. Sir.
Hello. Hello. Oh yes, I can hear you.
Sir, can you hear us? Hello. Okay. Okay, sir. Sir, there are some questions from the participants. One of the question is the safety ensuring mechanism is proven or not about the nanotechnology? Hello, sir. Hello, sir. Hello.
Hello.
Hello, sir.
Hello.
Hello. Hello, sir. Hello, sir. Can you hear us, sir? Hello, sir.
Yes, I can hear you. Can you hear me now?
Yes, yes, sir. Yes, sir. Yes, sir. We can hear you now. Yes, sir. We can.
Okay, so the question is whether the safety aspect of nanotechnology is fully addressed. That's what I believe the student you are trying to ask. Is it not? So I should say that most of those technologies that are applied have gone into, especially in the sector of electronics or consumer products, most of the technology has gone without respecting. Hello, can you hear me?
Yes, sir. We can hear you. Yes, sir. Yes, sir. We can hear you.
So I was answering the question. Yes,
Okay.
many of the
Sir,
technologies that have gone into the consumer products or into the electronic applications, etc., has not addressed the safety aspect of nanotechnology. But most of the technology that is gone into, for example, drugs or pharmaceutical applications, there is I would say like we know what makes them potentially toxic, and there are some precautions that are put in place. But carbon nanotube, for example, is used in many of those applications that I mentioned. But studies have shown that it can behave similar to asbestos that goes into our roofing, for example. Asbestos, as you may know, is a highly toxic material, especially for your lung if you breathe in, and that's because it causes cancer. So asbestos is banned from countries, Western countries, from as early as 1980s or even 1970s. But I think it is still being used in our place. But carbon nanotube has a similar morphology. If you look into the morphology, it can behave similar to asbestos. There is a phenomenon called as persistent fiber. If you inhale it, it can cause cancer. So now those kind of information has, I mean, like there were studies that address that before the mass introduction of or mass production of carbon nanotube. As a result, people have taken precautions to make sure that there is less or no exposure to carbon nanotube at a stage of production as well as at the stage of application. So there is minimal, or the risk is minimal. But with many other materials, especially these kind of materials that we encounter in food and agriculture, examples are silica or titanium dioxide, although there are studies done to understand their safety, there are not many studies done to understand how they transform when they are in the food supply chain. And that is some of the work that we are doing. So to conclude, or to summarize your question, whether it has adequate studies have been done, my answer is no. There are still lots of unknowns about the health effects of nanomaterial. But I would say, like, unlike many technologies that had been in use, nanotechnology has to some extent tried to address the potential adverse effect at an relatively early stage of the technology application. But there are a way to go. Yeah.
Sir, there was a second question. May I know how the nanosensors help for the detection of spoilage in food, especially if it is a packaged food?
So is the question how it detect?
Yeah, how it will detect, right?
Okay. There are several applications of nano in the detection of pathogenic microorganisms. So specifically pathogenic, you can actually, it's possible to actually see among a population of bacteria whether there is anyone which is causing hazardous outcomes, especially if you ingest it, or if they are going to cause spoilage of food. Both can happen. Okay. So one of the mechanisms of this, the way the sensor work is, you have, for example, gold nanoparticle which have surface receptors for the specific bacteria. For example, when there is a bacteria for which that sensor is made, when it encounters that nanomaterial, it causes a clumping, for example, or aggregation of this nanomaterial. As a result, you can easily detect by just measuring the UV absorbance, or maybe by even visual inspection, whether that food contains a bacteria, a spoilage bacteria. That is a simple mechanism, okay, that is based on gold particle, and that is manipulated in different forms. For example, a dipstick technology using platforms like a dipstick to detect the presence of bacteria and toxins using the same method or the same principle I mentioned. There are other possibilities. For example, the SRM, that is a surface resonance. Based on the surface resonance of the materials, you can look into if it is a fiber that is coated with nanoparticle, which actually, when there is with the interaction of light, it gives a specific range or a wavelength. But when there is bacteria deposited onto that, specifically designed for the kind of bacteria that you want to detect, there is a change in the surface resonance frequency, and you can detect it. So there are many kind of underlying technologies that enables the detection of bacteria, and now some of these technologies enable it to be embedded in the packaging material itself. So, for example, there are detectors for hydrogen sulfide. So you know that when the food putrefies by the action of bacteria, for example, it releases hydrogen sulfides. So gold-based nanoparticles can be used for the detection of hydrogen sulfide. And so after it reaches a certain level, for example, you will see a gradation in the color. So after it reaches a certain level, that product is no more consumable. You can throw it. So instead of using a expiry date based wastage or throwing of the food or discarding of the food, now you can look into a more of a detector-based decision making. Okay, so these are some of the examples. There are many other examples of nanotechnology for the detection of bacteria or microorganisms. There is another class of nanomaterial called quantum dots, which can also be used for multiplexed detection of bacteria, not one bacteria at a time, multiple bacteria, because you can use quantum dot with a different emission spectra in the same solution, and you can find whether the bacteria corresponding to that quantum dot that is functionalized to detect is present or not.
There is another question from Anakha, that is, if there is any application of nanotechnology in the machineries which can be used in food processing. I think
there is lots of application like food processing. There are a couple of applications. First one is the surface. I showed you some of the examples of how to make the surface super hydrophobic, for example, or super hydrophilic. There is an effect called the lotus leaf effect. You might have, if you have not seen lotus leaves by itself, you might have seen some of the images or photos of that, where even that, what's that called, some of those leaves that you might see when you just go to your backyard, so that these leaves actually doesn't get wet. You can see the droplets of water, just flowing as droplets. It is not actually wetting the surface. Lotus, although present in water, the surface is not wet, and that's because there is a nano protrusions on the surface. Basically, it makes the cavity air pockets. So if there is two nano pillars, you can imagine something like this, two pillars like this, and there is an air that is in between the pillar. As long as there is an air, the water cannot go in. So that is called as a lotus leaf effect. And that technology can be used, we can mimic that technology to make the surfaces more hydrophobic. And why is it important? You can avoid bacteria adhering or the adherence of any dirt or any materials that you encounter during the processing. Another technology is surface functionalizing the processing units with nano so as to minimize the bacteria, again, biofilm formation or any kind of spoilage microorganism adhering to the surface, especially with meat processing or milk processing units, etc. Another technology is using titanium dioxide again as an adsorbed material on the surface where you can shine UV light to get rid of the bacteria. So surface cleaning. Nowadays, you might even should know that there are buildings that are made with the glasses functionalized with titanium dioxide. So you don't have to clean that glass. You might have seen in big buildings, people climbing up and then washing the glasses, manually doing the work. And that's actually a skillful work. Nowadays, if you functionalize that surface of the glass with titanium dioxide, it is a self-cleaning surface. The dirt will not adhere because there is a constant removal of the dirt by the oxidation process that titanium dioxide when it interacts with the light. That's an example of surface cleaning. Obviously, there are other technologies, for example, nanotechnology to increase the dissipation of heat across for uniform heating of the surface or efficient heat transfer in food processing. So those are some of the technologies that are very specific for specific applications. So to answer your question, there are lots of similar kind of applications that are coming and also making the surfaces very strong. If you have visited food processing units like, for example, seafood processing, you know that there are trays that they use to freeze the fish with water in it. So after that freezing is done, what they do is they take the trays and bang it against a table to release the content. That, I mean, nowadays the technology might have changed, but what I am depicting to you is a picture of how a mechanical stability is important for those kind of materials that are used as a trays or the table. Otherwise, it will bend and it will be destroyed soon. So when the grains of that material, that is actually a sheet of, let's say, steel or some alloy material, that is not from a single crystal, it's kind of a polycrystal. If you look into the structure of that material, if you zoom in, you can see they are actually a collection of polycrystals, and each crystal is joined together. Crystal of iron or steel is joined together, and there is a boundary. So when that crystal size is smaller, that boundary becomes more extremely smooth. So the material becomes extra hard, so it won't break easily. So these are some of those applications of nanotechnology for food processing, where for mechanical strength, for improving the surface sterilization or cleaning properties, to get rid of bacteria on dirt, or to make sure that surface is more hydrophobic, etc. Are some of the examples.
Thank you, sir. Thank you for your answer. Sir, another question is, what about the legal rules and regulations for the application of nanotechnology in foods?
Very good. Excellent. So I'm really happy to take these questions, because when I interact with students, I know you are attending to my lecture. So really good. So that's an important question. If there is any regulation concerning nanotechnology application in food, there is no specific regulation that is very specific to nanotechnology application in food. There was some kind of push in European Union to regulate and to put that labeling for the use of nanomaterials in food, but unfortunately it did not work out. So as a result, you will not see, even if the companies are using titanium dioxide or silicon dioxide nanomaterials, you will not see those use of nanomaterials labeled. So by and large, there is no nano-specific rule, but there is carpet rules, carpet regulations that tells that any materials that are used in food should be safe. So for example, FDA does not regulate based on the size, but based on any adverse effect. If there is an adverse effect, they can take it out. But as long as there is no adverse effect reported for the nanomaterial, they can still use it in products. And that's why I mentioned specific case with the France, where they actually are taking out nano or titanium dioxide use in food. And that's because they found that from there is a group in France reported that it can cause neoplastic lesions in mice taking the nano titanium dioxide. So I don't think even in India they have even started talking about the regulation of, or even started talking about the applications of nano in food, although the industries are doing it. Okay, they are hiding behind the label.
Thank you, sir. And another question is that, what is the cost effectiveness of the mechanism in which nanotechnology which is applying to the foods? The mechanism of application. Cost effectiveness of the mechanism of application.
So that depends that depends on specific scenarios. So I mentioned to you, there are technologies that are very high end that requires more of budget and money to realize it its application. But there are technologies that are in the low end that can be used. So depending upon the scenario, if you want to, let's say, make sure that bioavailability of the material, for example, if you have a compound that you think in a spice or a condiment that you think is really important, and if we can nanosize it and we can make it bioavailable, because curcumin is the best example. Curcumin that comes from turmeric. There is lots of biological advantages of curcumin as an antioxidant, anti-cancer agent. There are lots of anti-infection, etc., etc. But curcumin, even in my opinion, the best remedy for viral infection, especially corona or COVID-based viral infections, the best remedy so far is the phytochemicals, chemicals that is coming from plants, like curcumin, or there is another chemical called quercetin. Quercetin is found on, for example, in ginger, and another one is piperin from black pepper, for example. All these are examples of phytochemicals. Now, many of these chemicals, although they are excellent, they are not available to the body. They are not bioavailable. So to make it bioavailable, we can make it into a nanometric form so that the body absorbs it. So if you eat, for example, turmeric by turmeric as such in your preparations, you may not absorb curcumin too much. You may absorb maybe 0.1% of what is going through the body, because curcumin is extremely hydrophobic. It's lipophilic. It may not be absorbed by the body. So if you are introducing a technology to make it bioavailable, you can go with technology that is high-end, like you can fabricate nanomaterials of curcumin, or you can low-end. for example, clay-based nanomaterials. There are some clays that can be used for introduction of materials, or even silica-based nanomaterials to improve the bioavailability, which are low end, like it's cheaper. So to answer your question, it depends on the kind of applications. So there are possibilities of developing. There are ways of developing economically viable technology for food and agriculture applications, of course.
Sir, another question is that does the food packaging material can cause a potential environmental impact? The food packaging materials, which is made up of this use of the nanotechnology, does this cause a potential environmental impact?
That's a great question. Like we talked about plastics, and we said that nanotechnology now can also go into these plastics to make it more preventive. It can prevent the oxidation or permeation of gas and moisture, etc., and also antibacterial. Now the question is this going to be more sustainable with respect to this end of life, what we call, okay, and after you use it and throw it? So the answer is you can make it either way. It can be more sustainable, less toxic to the environment. Who is that?
Sir, please continue, sir. Please continue. Sir, please unmute your. Sorry, sir. Please unmute your. Sir, please unmute.
Hello, can you hear me now?
Yeah, sir. Please. Now
Okay,
it's
so
audible.
I was talking about how to make nanotechnology for food packaging applications more sustainable. I was giving an example of cellulose. Cellulose is the most abundant biopolymer on Earth. So that can be used. There are nanocellulose. Cellulose in the nano size can be used, for example, for food packaging. In that case, it becomes more of a environmentally viable, environmentally sustainable, and less toxic material. But if you are using, for example, a nano-based polymer, for example, it may become even more concerned. Now, I mentioned to you, nowadays we hear about nanotechnology going into or nanomaterials of plastics in food and water. That should be concerning for all of us, because that is how we are exposed to these materials. So we don't want those kind of technologies, especially when I talk about sensors. Some of the sensors that are used for detecting the pathogens, for example, quantum dot, they are not really safe. They are actually made up of chemistry that are toxic. For example, cadmium. Cadmium selenide makes the core of quantum dot, which give different colors. For in that kind of scenarios, it is not very sustainable. It may actually add on to the burden of what is already there. Okay.
Okay, sir. Okay, thank you, sir. Then another question is that what are the difficulties faced to detect the hazards caused by nano devices and the nanomolecules in foods?
That's also a great question. So there are not much of methods and tools to actually detect, even quantify nanomaterials that are applied in food. That is one of the difficulties in its regulation. So if you want to regulate, you have to make sure that you can detect it, if somebody is using it or not. So the tools and methods for detection is at its infancy. So there is room for improvement there, but there are a couple of new technologies that are coming up for the detection. So to answer that question, it needs improvement, but it is in the pipeline. The technologies or the methods are in the pipeline for the detection and detection of also of the risk of nanomaterials. Okay,
Thank
so
you, sir.
I will
by
take
a conclusion,
I'm sorry.
Yeah, by a conclusion, from your point of view, when we can expect this technology in our country, in India?
In India, I mean, if you are consuming food products, it's already there. I mean, it is not the government that brings the technology. It is the industries that enable this technology. So the industries are smart. They are using it already. you are, like I said, like you are exposed to titanium dioxide or silicon dioxide or even nanoparticles of silver by being a consumer of those technologies. So it is not, I mean, what is distant, what is not being applied is, for example, aquaculture or farming, the use of nanomaterials in farming. And I believe there are opportunities, and there are some companies maybe they're already operating with products that goes into the farms, like for vegetable farming as well as animal farming. So I was mentioning to you before, I have 2 more minutes and then I have to run. So I may not be able to take more questions, but I will be, you may feel free to shoot me an email. And you can find me from Google or from, you can get my email from Minimis if you have more questions. And when I have time, I will be able to answer those questions.
Okay, sir. Thank you. Thank you for your valuable words. And as a conclusion, we are concluding from our HOD, Ms. Minnie Michael.
Okay.
Hello everyone. It was one of the rarest webinars conducted in the field of nanotechnology with lots of information. So as we have seen, many of the students are keen in having about this technology from this in the field. We have requests from many parts of the country to join this webinar, but unfortunately, we have limitations of Google platform. We are unable to process the request. This reveals the success of the program. This program was live on the YouTube with many viewers, and we can provide these valuable lectures to the interested persons. So I thank you, Dr. St.George, from the part of St.George College, Arvitra, Department of Food Science and Technology, for spending your valuable time with us. Thank you, Sajid.
Thank you. It was my pleasure.
Thank you all the participants, especially beloved Alapet sir, Principal Reggie sir, Bus sir and course coordinator George Pillagalai, their teachers and students. So I would like to thank once again. Thank you very much.
Thank you all. It was a good show.
Yeah.
Thank you, sir.
Yeah,
Thank you for attending. I will try to reach out to you. So, Minimisinde Sunday email ഒക്കെ ഉണ്ടോ?
I mean, I will hand it over to her,
Okay.
See, when you get time, whenever you come out, when you come out here, please pay a visit to the college and to us, okay?
Sure.
It was a very, very, very informative lecture. A very, very good one. Okay? Thank you very much.
I didn't go into the details of
thank you all. Thank you
studies.
very much.
Okay. Thank you.
Thank you, sir. Okay then. സാറിന്റെ കൊച്ചുമോളാന്ന് തോന്നുന്നു.
Can you see her?
Yeah. Hello.
Yeah.
Hello. What's your name? മോളുടെ പേരെന്താ? Nita. Wonderful.
Yeah.
Thank you.
We are the close friends,
Thank you all once again.
We are the close friends.
I see. Okay.
Okay. We talk in the same wavelength. We work in the same wavelength.
Oh, wow.
Okay, sir. Thank you very much.
Okay, sir.
Take care. Take care.
Yeah.
All the best.
Thank you, sir.
Good day. Good day.
Bye-bye.
Some